So conventional MRI such as T1 weighted and T2 weighted images have been used to characterize and grade glioma tumors. They're good at determining the location of the lesion, the mass effect and necrosis of the tumor, and the differentiating edemas in the brain. However, they have been shown to mistake high-grade tumors for low-grade. And furthermore, conventional MRI techniques does not take into account other tumor physiological factors such as microvascularity, angiogenesis, micronecrosis, and cellularity that are important in terms of the grading. And so diffusion MRI can be useful in determining in classifying the grading and the early detection of gliomas.
So diffusion MRI basically measures the rate of water diffusion at each voxel. And to go about doing this, technicians will usually apply a large gradient in the XYZ direction, as diffusion is in all directions, on top of their initial pulse sequence. So they're basically making the signal very, especially sensitive to diffusion. And to get this, the signal amplitude depends on three factors. The first is rate of diffusion. So the faster the rate of diffusion, then the more the different gradient field strengths that the molecules will experience, and therefore the more out of phase that they will be. And the longer we wait to observe the signal, there's also more time for the molecules to diffuse and also go out of phase. Similarly, for the strength of the gradient magnetic field, so the stronger it is, then the more the molecules will dephase. And so the first feature is what we are trying to observe or detect, while the latter two are are parameters that we control. And as we see on the right image, the molecules that are truly stationary will rephase after the 180 pulse. However, those that are diffusing will dephase even after the 180, and therefore you will get attenuation of the signal. So to analyze the diffusion weighted image, we want to refer to the B value, which reflects the gradient amplitude and duration of how long the gradient was left on. So if you have a high B value, then you either have a high gradient amplitude, or you left it on for a long time, or both. So to compare the original image with the diffusion weighted image, we have to refer what is known as an ADC map. And basically this map quantifies the speed of diffusion, and it avoids T2, what is known as T2 shine through. So if we look at just the diffusion weighted image, we see a very bright signal where the glioma is. However, just by looking at this image, we don't know whether there's actually restricted diffusion or if it's just the very high T2 signal carrying over into the diffusion weighted image, which is known as the T2 shine through. So to reveal the truth, we need to look at the ADC value, which basically uses this equation right here to calculate the values. And by looking at the ADC, you are able to tell whether exactly there is actually restricted diffusion or not. And as we know, tumors have varying degrees of tissue density. They could be hypocellular or hypercellular. And so we can take advantage of this subtlety to detect early signs of pathology. And on the right, we see that there is different grades of tumors. There is, so the higher the grade, the more dense the tumor is. And this means that there will be decreased water in the interstitial spaces and therefore restricted diffusion. And so the high grade tumors, you will get a high signal on your diffusion weighted image because there's restricted diffusion. So then the molecules will have less room to diffuse and therefore go out of phase. And then therefore you will have a corresponding low value on the ADC map. However, for lower grade tumors, which are more hypocellular, you will have more water in the interstitial spaces, and therefore you'll have more diffusion and lower signal on the diffusion weighted image, but higher value on the ADC map.
So to give you a visual representation of what Rosie was speaking about, we have the T1 and T2 weighted images, the DWR image, and the ADC map of 3 grades of gliomas. A represents grade 4, B represents grade 3, and C represents grade 2 gliomas. So with T1 and T2 weighted image, it's possible to talk about the size of the tumor and where it is localized. Whereas the information that DWI and the ADC maps give you is the vascularity and the hypercellularity of the tumor itself. So like we learned in lectures, in case you have a tumor present, depending upon the cellularity, the diffusivity is going to decrease, and hence the signal intensity is going to increase. This is denoted by the red color arrows that mark the regions of high signal intensities. And so this can give you potential information about the cellularity of the tumor. And this has been used typically in medical practice to grade the tumors. So what we propose could be an application of DWI and ADC is not just using DWI and ADC for grading tumors, but also to diagnose them in their earlier stages, because it gives you the changes in pathology way ahead of the conventional T1 and T2 weighted images. So since ADC will give you the diffusion coefficients of the tumor in its various spatial orientations, we can use it to quantify the pathological changes in the brain as well as tumor cellularity. This early diagnosis would allow us to chart a treatment course for patients, because these tumors often occupy vital areas of the brain. And by the time we diagnose them, they already have a poor prognosis, and they're already in a stage of progression where we cannot treat it or save the patient. So how do we calculate these ADC maps? So we really spoke about a bit of what the equation that we use to get the diffusion weighted image. So I'll go in a little bit more depth of how these ADC maps are calculated. So biological tissues are typically anisotropic in nature. They have different diffusion coefficients that vary depending on the direction on which you measure it. So we make use of this property in order to calculate the signal intensity along the different directions, and we consolidate it together in order to get our diffusion weighted image. These diffusion coefficients could be represented by a diffusion tensor, which is basically a 3x3 matrix. We take the average of the trace of this matrix in order to calculate our apparent diffusion coefficient. The trace of the matrix is just the sum of its diagonal elements. And so taking the average of that would give you the apparent diffusion coefficient. Another way you could calculate ADC is by dividing the diffusion weighted image by the baseline image that we take at every single B0 value and taking the logarithm of that in order to calculate the apparent diffusion coefficient at that point. When you consolidate all these values together, you can chart out an ADC map.
All right, for our Matlab demonstration, we use the DTI and fiber tracking toolbox to generate this ADC map. So the way this works is we first obtain our S0 and S1000 signal. So S1000 is basically the signal at the DWI value, the DWI image. And then we calculate the voxel, we converted the voxel intensity to absorption. So that's basically taking the natural log of S1000 over S0. From here we only process the voxels that were above the background threshold, which we can decide what that is. And the next step would be to calculate that diffusion tensor that Vaishali spoke in a previous slide. So that's 3x3 tensor. And this is basically solving a system of linear equations. And from there, once we get the 3x3 diffusion tensor matrix, we can calculate the eigenvalues. So we'll get 3 eigenvalues there. And before we compute the average, we'll want to regulate this. And the reason we want to do it is if we do find any negative values, that could be due to noisy image acquisition. So you want to kind of regulate that. And then getting the average gives us the ADC value of that specific voxel. And then we can repeat this process for each voxel. So in this slide here, we kind of see the outputs from our toolbox earlier. So on the left, we have the diffusion tensor images. So the diagonal elements the intensities are pretty similar and don't change too much. So the reason why we don't see a dyx, for example, is the dxy and dyx, or there's a symmetric matrix there. And to the right, you can kind of see how the ADC map is almost looks like an inverse of the DW image there. In conclusion, we can kind of see how ADC maps are helpful in grading gliomas. So now, so far, we've kind of used them to grade gliomas, and hopefully, we can start using them to diagnose early stage gliomas as well. So on the left, you can see, depending on the type of grade, that ADC histogram kind of changes. And to the right, we can just see that the ADC histogram from our toolbox here. That was from just a sample data set. It didn't have a claim actually, because we couldn't find, we didn't have the adequate data for that. And these are references. Thank you so much.
Great. Any questions from either online or in the room? Okay, we have a minute, so let me. So could you clarify a little bit, sort of what data did you actually use? Did you actually have glioma, like the figure 7? Is that your figure or?
Yeah.
So figure 7 is from paper that we use because we were figuring out what can we do with ADC values, and we saw that they use it to grade. So that's what we didn't have. So what we show on the right is what we got with our like an example data set. So it was just like an image of a diffusion weighted image of the brain. It didn't necessarily have a grade 1, 2, or a glioma.
Great. So that might be like, you're saying that's maybe from a normal brain.
Yeah.
Okay, great. Very nice.
Yeah, we didn't have just kind of data for
Sure. Okay, well, great. Nice job. Any other questions? Okay, thanks. So the next group is the MRI microscopy group. Do you guys want to come on up?
Hi everyone, I'm Hona. This is Joey, this is Ryan, and we're joined by Mabel online. And we're here to talk about MRM. This technology is called magnetic resonance microscopy. And this is a very high resolution technology for MRI. And right now it's only for research purpose, not clinical. Okay. So Mabel, if you are good, you can start it.
Okay, can you hear me properly? Okay, so I will start with an intro review of MRAM, basically non-invasive imaging technique. Hello?
Oh, we can hear you, yeah.
Okay, based on spatial encoding of NMR signal to form images of optically opaque objects with a spatial resolution in the micrometer range, so less than 100 micrometers. It runs on the same fundamental physical principles as MRI. However, key differences to note is that it's basically a higher evolution of MRI. It has stronger gradients of around 50 to 100 times greater for spatial encoding. Medical MRI resolution is around 1 millimeter, while MRM resolution ranges from 100 to 10 micrometers. The voxels are up to 100,000 times smaller, but this means that the signal is also up to 100,000 times weaker. MRM chambers can typically be around 1 centimeter cubed, and the applications of MRM extend far beyond just diagnosis, like with an MRI. So if you look at figure 1, go back. You can see the scale of magnetic resonance microscopy. On the left is a 5 millimeter slice of human brain next to a 40 micron MRM slice of a mouse brain. And the size is depicted by that little white box. And the spatial resolution in the coronal plane is around 1 by 1 millimeter compared to in the mouse brain, 40 by 40 40 microns, meaning the volume is nearly 80,000 times smaller. And on the bottom right is a biospec MRM scanner manufactured by Bruker, but this one actually has a larger bore for studies of mice and rats. Next slide, please. Much of the success of MRI comes from its high sensitivity to pathologic alterations. So when we go on the microscopic scale, the applications are wide and robust. MRM can be used for histology, vivo cell tracking, restricted diffusion imaging, functional magnetic resonance microscopy, and three-dimensional mouse atlases. MRM can also be applied outside the field of biology with fluid mechanics, geology, and chemistry. MRM can measure flow and diffusion, so it can be applied to characterize porous media. Included on the right are some images generated by MRM. Figure A and B on the top right show remotely detected images of gas flow in a microfluidic device using hyperpolarized xenon. The figure below shows a hypocotyl of a 6-day-old castor bean seedling showing plant anatomy with cellular-like patterns. And the figure on the right shows a neuroatomic MRM image of a mouse brain. Next slide. While the applications of MRM are broad, one of the most valuable capabilities of it is the ability to image small live animals. Compared to conventional histology, the subject can be viewed interactively in any plane. Spatially localizing morphologic and functional changes in the organ systems of small animals means that we can get a better understanding of things like embryonic development, genetic mutations, potential therapeutic treatments, and the effects of environmental insults. In addition, the capacity to perform repeated non-invasive imaging in the same animal enables us to study the longitudinal progression of a disease or treatment. This also means that there's a reduced need for animal sacrifices, and overall, MRM is a promising platform with broad applications in biomedical research. And you can see in this figure the scan of a live rat, and you can see all the organs stained for clarity. Next slide. There are some barriers to MRM, however. General limitations include the fact that for small animal studies, radio frequency pulses and gradients used for imaging distort electrical recordings, such as those for cardiac monitoring. They are also at a greater cost to maintain, and achieving maximum resolution can take many hours, which is impractical. Compared to other animal study methods, animals in a bore require remote physiologic monitoring and delivery of agents for the study, which can complicate things. Overall, relative to clinical MRI, MRM poses many more additional challenges. There is a substantially weaker signal in comparison to MRI, and this is due to the fact that fewer protons occupy the much smaller voxels. The fundamental resolution limits are defined by effects that are not as important in MRI, like diffusion and natural line width, as well as the exacerbation of motion artifacts during in vivo studies. Signal to noise ratio is the greatest factor and major obstacle when trying to attain the highest spatial resolution. Next slide.
Okay, thanks, Mabel. So now I'm going to talk about some limitations of MRM and how we are going to improve it. And first, as Mabel have said, the MRM has a much higher resolution than clinical MRI. It's like this photo shows. You can see there's a tiny rectangle box compared to a human brain, but it's a mouse brain. So according to what we have learned in this course, a very intuitive hunch is if we increase the gradients, the resolution will naturally go up, right? So here we use the point spread function and also phantom to show as the gradient goes higher, you can see the resolution goes higher, so we can tell if one point spread function can be distinguished from the other, and also the border of the phantom and the organs of the phantom can be distinguished. And also there is another very important factor in MRM, which is called CNR. It's contrast to noise ratio. And it's very similar to signal to noise ratio, but it's kind of different. So as you can see in the first two images, this is T2 star and T2. So you can see a very faint, brighter phase-shaped line here. This is granule cell layer, which you cannot see in the diffusion image. But in the diffusion image, you can see a dark appearing band here, but you cannot see in T2 or T2 star. And it's multi-fiber pathway. So apparently, because they have different contrasts, so that we can see different organs in it. So one way, but we can only scan for a certain amount of time. So there are only a certain amount of sequence that we can apply to generate different contrast. So after we got the image, we can actually use post-processing to improve the contrast so that we can see different organs. But this will naturally cause problem. So this is a brain organ that we add noise to it, and we do a post-processing to enhance its contrast. And you can see clearly that CNR is rising. And it is defined by the variance of signal over the variance of noise. And also, if we increase this CNR, some signal from the organs will disappear naturally, but the noise, they keep the same intensity. So the SNR will naturally dropping. The SNR here we shows as a mean of the signal and over the variance of the noise. So this is a kind of big trade-off. You want to higher CNR or you want a higher SNR. And so there are two things that we need to consider. And next, I'll pass to Joey so he can talk more about this SNR trade-off.
Yes, so the relationship between the spatial resolution and the SNR is very similar to the relationship Haonan introduced on CNR and SNR. They are also inversely proportional. So on the left, you can see that just to simplify everything, we just assume the noise is white noise. So on the left, you can see that there are like two images reconstructed using different matrix size. The larger the matrix size, the more higher frequency K-space samples we're taking, therefore we're getting higher resolution. So the image on the top left is an image with like not that much of a noise in it. And as we can see that as like the matrix size decreases, the image blurs a little bit and the SNR increases. But overall, for the top left image, it doesn't seem to be affected that much. However, for the bottom left image, you can see when the matrix size is very large, the image is almost a black box. But when we gradually drop the matrix size, the image becomes a bit more recognizable again, but most of the details will become obscured if we decrease the matrix size. And then similar result is also shown in a 1998 study demonstrated by the graph on the right. So in this case, they are using voxel size to represent the resolution. So the smaller the voxel size, the higher the spatial resolution is. And as we can see, when they are increasing the voxel size by decreasing the spatial resolution, the SNR increases. However, the caveat is that for their cases and for the simulation we produce are for generic MRI. And in their research, they concluded that even at a 22.4 SNR with facing the higher spatial resolution, they can still produce reasonable diagnostics on small joint and lesions. But for MRM, the stuff becomes a bit more difficult. First, we need to know that the noise in the MRI system, in the MRI system, is caused by basically random motions of the electrons, and it's kind of hard to reduce that. So, and then as Mabel and Haonan had introduced in earlier slides, the resolution required to achieve magnetic resonance microscopy is way higher than that of the traditional MRI. Therefore, in order to counter it, therefore we simply cannot play the trick of like balancing between SNR and resolution, because once we balance all that, we simply can't get the other one to be workable. Therefore, we need to find another way to improve the SNR. One way would be to improve the signal strength. And then in order to do that, we can do so by using better coil designs or other hardware changes, which Ryan would introduce a bit more on that topic.
Just to give a better perspective on this decrease in signal strength, these boxes on the left show the voxel sizes compared to one another. The large clear cube is 1 millimeter isotropic, which is the similar size to classical MRI, where the blue cube is 100 micrometers ISO. And then if you look in the very center, it almost just looks like a small dot, is a cube that is 10 micrometers. So as these sizes are getting smaller and smaller, there's way less protons, which means you're going to get a much weaker signal. And there is no way to design around that problem. You can't change the amount of protons inside of a specific type of material. So to look at signal strength, the equation that we're using here is the induced EMF inside of the coil. While there are some things you can do, like changing how you're amplifying the signal or your A to D converters, this is the best way to view the actual hardware that we're using. So as you can see in the induced EMF equation, there are a couple factors that we can consider, like the magnetization and the Lamar frequency, which are both coming from your main magnet. So just as in similar to your clinical MRI or classical MRI, you want to have the strongest magnet as possible, but you can only make it so strong. So the other option that we can change is our B1 field, which comes from our RF coil. So we're going to look at how coil design affects your RF magnetization field and how we can optimize that to be able to get a stronger signal, which helps as we go down, or we get better and better resolution, smaller size. We're able to have a higher sensitivity to whatever we are trying to image. So in these 3 graphs, we are showing the signal strength coming from these coils. A very important concept when looking at coil design for the situation is considering a theorem called the MR Reciprocity Theorem. For anyone who is familiar with antennas, you have probably heard of the antenna reciprocity theorem, which allows you to calculate how well an antenna is able to pick up a signal by understanding how well that antenna is able to transmit the signal. It works very similar in MR systems, where your coil's ability to receive a signal is going to be proportional to that of the coil's ability to send out the signal. So that's why we can look at the strength that these coils give and understand the sensitivity that we are going to get back from them. So the first 2 coils to look at are the birdcage coil and solenoid coil. Okay. Which, as you can see, as the radius of the coils gets smaller, they get stronger. So basically what this says is we want smaller coils. So as we're designing the coils, we want to keep that in mind. And then also the second one to look at is surface coils, where essentially, while they also have the similar design, it actually depends on how far away from whatever you're looking at you can get to optimize that strength. But one of the great things about MRM systems is that they are designed for you to be able to trade in and out coils. This is why RF coil design is so important with MRM. They have these beds where your test subject is on, and you can swap out the different design of coils. All right, are there any questions?
Great, any questions from the room? You guys? I had a just turn off your volume. Yeah, I had a question. So could you go back a few slides when you're looking? You had that slide where things like SNR and spatial resolution was changing.
Yes.
Could you just explain what is going on the top row and what's going on the bottom row?
So actually, the top row, essentially the top row and the bottom row, they are identical. It's just for the top row, I believe the noise magnitude we're adding in less than 1% of the signal strength, but of the image signal strength, and for the bottom one, I believe that's noise we're adding something like 10% to 50% of the noise.
I see. So they're just 2 different SNR regimes you're trying to describe. And so then when it goes black, that's when it's high resolution, or?
Yeah, that's when it's having its maximum resolution.
Okay.
With all the noise in, so the image is kind of like.
But would it? I mean, okay. So if you scaled it, it wouldn't necessarily be black. It would just be very hard to see, right? I mean, you're just auto scaling it, right? You're keeping it on the same scale, so it looks black on that scale.
Yes.
Okay. Okay. All right. Thanks. Any questions, Dr. McVeigh?
I don't know, but I have seen 2-micron isotropic, the smallest I've seen.
Wow, that's 2 micron. That's pretty amazing. I guess people are getting to like single spin stuff, right?
Yes, so there's
So-
magnetic resonance force microscopy,
Okay.
which uses, instead of an antenna, it's like a little needle with a magnet on the end.
Right. Right. Okay. That's pretty cool. All right. Great. Good job. Okay. So our next group is T1 and T1 2T weighted imaging. Can you stop sharing your? Yep. And are you guys doing it? You're all online, right?
Yes.
Yes, we are.
You able to see the screen?
Yeah, we can see the screen. Go ahead.
Naomi?
Yes, I'm here.
Perfect.
All right. So I'm Naomi Wilcox, and my project partners are Abe and Jimin. And today we'll be talking about T1 and T2 weighted imaging and MRI. So a quick content at a glance. So we're going to just quickly discuss the principle of MRI, some brief background on T1 and T2 weighted imaging. Then we'll show you 2 exercises in Matlab for these various weighted imaging, and then also introduce a new research-based method people use called T1-Row. Okay, so talking about the quick principle behind MRI. So we know that it's often used in clinical practice because it's non-invasive imaging technology, often used for detecting disease, diagnosis, and then monitoring the treatment of clinical patients. It's a sophisticated way also to just measure the change in direction of the rotational axis of protons, which make up living tissue. And so figure A here shows Diamond is a part of a lab that works on MRI in mice. So that's just him at his lab. And then if we look at the figure B, this is a quick video with we've worked on in homework showing a magnetometer exercise. So as we play this video, as you can see, when we start out behind the yellow dash line, the value is around 1 Gauss, which is close to the OSHA limit for 5 Gauss. But as we move closer in toward the MRI machine, we reach about 30 Gauss. Okay, next slide. All right, so this is just some definitions of T1 and T2 weighted imaging, but essentially they recognize the various relaxation times for different tissues. So at the 3rd bullet, we see that an excitation pulse essentially rotates the magnetization vector away from its equilibrium state. So this results in vectors that are longitudinal, so the MZ axis, and then transverse MXY components. And so T1 and T2 are different spin lattice and spin time constants in order to return to the equilibrium of their specific axis. And so on the right, figure A, this is another video, essentially just showing the various signal that's resulting in changing the TR and TE values and compared to the amount of time it takes in order for these fat and fluid tissues to become dark or bright, depending on what we're looking for. All right, next slide. Okay, so yeah, on the left we have figure A, and this is just an image of the weighted T1 and T2 weighted images of human brain. So below we have the table 1 with parameters that we use for TE and TR. So you can see that this CSF changes in brightness as we vary these parameters. And on the right, these are images from, I believe the Morris Cancer Center is the laboratory I mentioned before. So it's an image of a mouse brain. And below are the parameters we used for the T1 and T2 weighted image. So as you can see, the total time for the T1 weighted image is around 45 seconds. versus a little bit longer, around double it's around 9 seconds for the T2 weighted image. Okay. So this last one is just, we've seen this equation before, but it's producing the signal using the partial weighted images. So on the left, we have the T1 weighted image. So we can see that here we're fixing a TE value of around 1 millisecond. And as we vary the TR, the contrast increases. And then on the right, figure B is a similar exercise, but here we're fixing TR to 12,000 milliseconds, and we're varying TE to get different contrast. All right, next.
Okay. Yeah, so now we'll talk about the T2 star relaxation in MRI. So T2 star decay refers to the exponential decrease in the signal strength following in the initial excitation pulse as a function of time, constant T2 star. And we studied in the lecture that the distortions in the magnetic field can be generated by the deoxyhemoglobin, and also sometimes the metal implants and other contrast agents. Figure A here is a MRI from the 17-year-old male that we can see it's a glioma diagnosis. But when the image is the T2 star weighted, we can see more hemorrhaging around the glioma, right? So that was one useful benefit for the T2 star weighted images. We'll quickly talk about the flip angle. So now we already know what flip angle is. It's the hydrogen proton shifts from its longitudinal plane to the transverse plane. And flip angle is important, and why it is very much important when you're talking about the T2 star weighted images. I'll just quickly talk about that. So figure A here, we can see it's a T1 weighted spoiled GRE. And over here, the flip angle is about 30 to 50. But when we reduce the flip angle and made it about 5 to 15, it became the T2 star weighted image, and also by changing the TR and TE. And what is this spoiled GRE? I will talk about that next. So spoiled GRE is a spoiled gradient echo sequence. It's one of the typical sequence which is used for T1 weighted images, and as the T2 coherences are disrupted, but it is also highly useful for producing the spin density as well as the T2 star weighted images. So over here, we can see that in the gradient spoil sequence, we ignore all other imaging gradients so that we left with the RF pulse and then the spoiler. So this is the spoiler signals over here in the gray in the boxes, right? Now, over here, it's just a little animation for the tip magnetization, which is the 1 cycle. It rotated 1 cycle for the dephasing. Whereas then when we talk about for the second figure over here, which will appear. So over here, we have the repeat tips vector with the different colors. And that's repeating the sequence over and over for the tip magnetization. And then that will at some point reach to the steady state. So as you can see, that slowly it is trying to reach towards the steady state. And then so the steady state SS will be formed. Right, and then this SS is actually a familiar SS because the magnetization has distribution within the plane before and after the RF pulse. So you see here is the RF pulse. So here we can see that the before and after RF pulse, it's in an elliptical motion, it is moving around. And also one thing to keep in mind that the gradient spoiler does not eliminate transverse magnetization in the steady state. Okay. All right, so just little bit summary to summarize what is this spoiled GRE is. So again, we have the RF pulse, we have the spoiler over here, and this is the signal. And then each spin has a different precision, and the average balance of the steady state free precision, which is SSFP at start of the TR. So when if you see that the peak of the TR, we see at the start of the TR, and then it's also crossing over the peak. And also it can result into the some T2 decay because of the TE from RF pulse to the echo time. So this is like overall a spoiled GRE is. And this is the equation that we used for our Matlab exercise, which I'll talk about next. So over here, the S is the signal strength, H is the spin density, K is the scaling factor, and alpha is the flip angle. And this term we saw in the spin echo equation when the professor taught us in the class. And of course, we know that when the TR increases here, it's becoming more T1 weighted image. But over here, when the TE becomes longer, it becomes more T2 star weighted imaging. And the highest signal strength is achieved when the alpha E, when the alpha flip angle becomes alpha E, which is the Ernst angle. And over here, I just tabulated some parameters which you can go over later on once the slides are uploaded. So basically what parameters we should use in order to get the T1, T2 weighted image, T2 star weighted images. And basically the importance that I'm highlighting is the flip angle over here. The flip angle is also important along with the TR and TE. This is the Matlab exercise that we performed. And over here, it's a 3D plot. So what we did that we used this T1, T2, and T2 star values from the literature for the gray matter and the white matter for the brain. And then over here, we did the three-way relationship between the echo time, the TR, and also flip angle. And basically what this plot is showing over here is in order to achieve the best gray matter, white matter contrast, you need to have certain TR, certain TE, and certain flip angle. And then that's a three-way relationship over here. And just to give one example here that that's at particular this TE and TR, at this point, the flip angle is 46.13.
Okay, so this is where I'll explain what T1 row is in MRI. So as opposed to their conventional spin echo sequences, T1 row involves first a the magnetization of the longitudinal axis. And then after that, we do have a refocusing of the magnetization of on the transverse axis with this RF pulse, which we will call which is called the spin lock pulse. And then after that, the longitudinal magnetization is restored with another RF pulse. So in total, we have 3 RF pulses as shown in D, where we see a 90-degree in X, the spin lock pulse, which is dictated, the time is the TSL, and then the third RF pulse, which restores the magnetization in Z. And then afterwards, we have a normal imaging pulse sequence to allow us to obtain the images. This sequence essentially gives us T1 row weighted images. And the equation on the right over here dictates the signal based on T1 row. Next. So to distinguish T1, T2, and T1 row, in the image on the left, we see T1 relaxation is based on the restoration of the longitudinal magnetization on the Z-axis, where the magnetization is shifted to the Y-axis, and then the time that restores back to the Z-axis is the T1. T1 is very long in water, for example, and very fast in fat tissues. And that's why we have brighter signals with T1 weighted images in the fat tissue compared to the cerebral spinal fluid. T2, however, in C is the decay of the magnetization on the XY axis caused by dephasing of the spins. And fat tissue, we have much shorter T2 compared to water. So therefore we see fat appears darker in T2 weighted images than the cerebral spinal fluid. To explain T1 row, what happens is that we have a RF pulse in the Y axis, and this a rotating RF pulse around the Y axis. And what happens is that we see precession in the ZX plane. And what happens is that this causes a refocusing of slower spins caused by slower macromolecular tumbling, which is called spin locking. And in images A, B, C, D, and E, we see the evolution of the refocusing of these spins. And this allows us to get better contrast based on the kinetics of larger molecules, such as proteins, as we will show in the next slide. So one application in research for T1 row MRI is looking at is diagnosing osteoarthritis. So although T1 row was described back in 1950s, it's actually become a very new hot topic in clinical research, where T1 row mapping is used to evaluate the degradation of cartilage in the joints. What happens is that you have a very short T1 row when the cartilage is intact, but then you have a very long T1 row when the cartilage is obliterated. And what happens is that when the that you increase the hydrogen exchange in native proteins versus immobilized proteins in the intact collagen. So therefore you get very long TR as a result of the collagen network being broken, as opposed to it still being intact, where the T1 row will be very short. Therefore, we will see, we can distinguish the kinetics in proteins using T1 row. All right, and that's the end of our presentation. We wanted to acknowledge Dr. Liu and the TAs Ji Yuan, Yi, and Junming Zhang, and the Department of Radiology at the Moores Cancer Center for allowing us to image those mice. Any questions?
Thank you. Great. Any questions from the audience? No. Okay, so so what sort of in terms of the time constants of T1 row, what are the typical time constants, or what sort of, is there a typical range of T1 row values?
Typical range of T1 row values are similar, are on the, I would say, on the order of magnitude of T2, because we're still dealing with relaxation in the transverse plane. So the time constant we would use is the TSL, which is the times, which is the spin lock time. And these times can be on the order of 80 milliseconds to 100 milliseconds for, what was it? For, I'm sorry, I don't have the example for the, for any TSL.
Okay, yeah, because I think with the advantage of the TSL is you can have those spins locked for quite a lot longer than the T2. So that is an interesting property of T1 row.
Yeah, they are, if I read correctly, they were actually slightly longer than the T2s.
Great. Okay, anything else? All right, thank you again, and we'll move on to the next talk. So let's see, the next talk is going to be on FMRI analysis using Combat. Is that in person here?
Yeah.
It's a hybrid, okay. Yeah, we're hybrid. Great, all right.
All right, hi everyone. So our project is FMRI analysis and harmonization using Combat. My name is Chloe and my partners are Derek online and Michael in person. Okay, to start off, I'll be giving a brief recap of what FMRI is and how it works. Of course, we know that functional MRI allows us to understand what the brain is doing. So rather than focusing on spatial resolution like an MRI, we prioritize temporal resolution in fMRI and try to get on the millisecond timescale in order to quickly capture neuronal activity. Statistical analysis is then used to determine which parts of the brain have significant correlation to the stimulus presented. The first fMRI studies were done in the 90s, in which they compared brain activity when a patient looked at light versus dark, and found a lack of activity in brain regions that were unrelated to vision. Now, a huge number of fMRI studies are done to characterize the relationship between the brain and drugs, sleep, food, and more, as well as understanding the functional connectivity of the brain in different states like development, aging, and neurological disorders. So here are some of the equations that Professor Liu taught us in class about how fMRI works. We learned that depending on how much inhomogeneity and dephasing there was, this would dictate how much MR signal we would get. This R2 star relaxation rate is also related to the bold signal change, the blood oxygen level dependent brain activity, which is the difference between the activation and baseline signals divided by the baseline, which I'm going to delve further into on the next slide. So oxygen is attached to hemoglobin in your lungs, and these hemoglobin molecules carry oxygen to your brain. In the brain, the oxygen is taken off, forming deoxyhemoglobin and leaving 4 iron atoms that perturb the magnetic field. The amount of deoxyhemoglobin in your vein is equal to the cerebral metabolic rate of oxygen divided by the cerebral blood flow. Our brain has figured it out such that the blood flow increases much more than the rate that oxygen is used up, which overall decreases the concentration of deoxyhemoglobin, which in turn decreases dephasing, decreases the R2 star value, and increases MR signal. So here's what the fMRI setup looks like. It's similar to that of MRI, but you're asking the patient to perform a task or play a game on the screen. The data output consists of 3D anatomical and 4D functional scans, which are analyzed collectively. There are many parameters that can be set on an fMRI scanner, but I'm just going to go over a few of them that are going to be relevant to our project. The echo time is the time delay between excitation and acquisition, and the repetition time dictates how frequently the nuclei in a particular location are excited. If they are excited much more rapidly than the relaxation rate, the protons will not recover to equilibrium in between the excitations. Both echo and repetition time control the contrast of the acquired data and the amount of signal that can be encoded. For phase coding steps, in order to localize protons, we apply separate phase encoding gradients for each row of pixels. If an image is 256 by 256, we would have 256 phase encoding steps stored as different rows in the k-space matrix. And lastly, pixel bandwidth is the range of frequencies represented in an image. If the bandwidth increases, the number of hertz per pixel also increases.
Michael, are you there? Having trouble hearing. Michael, I'm not sure if the Zoom participants can hear what you're saying.
Talk loudly into here. You come over here then.
So variability in FMRI study. So a study by Glover and Al in 2006 identified the fact that in multi-site fMRI studies, they had a median inter-site variability and localization of a ROI by 6 millimeters, which was considered significant as opposed to inter-site differences when doing the study. These were a study of the same subject, image at multiple different places, performing the same exact task in order to make a control for it. And the question they asked themselves was, what is leading to this difference? What's leading to variability here? Variability they found, and that they most thought would be the biggest source of the change, had to do with the K-space data acquisition between the two different sites. So the big thing about the two sites, they're both 3 Tesla scanners. However, one of them was a Siemens and one of them was a GE. The GE utilized a spiral out K-space imaging, whereas with a Fermi filter, which we have here, and the Siemens used a traditional echo plane gradient with a Hamming filter. The difference between the Fermi filter is that we have a Fermi radius, which is kind of the bandwidth. Frequencies that lie inside the Fermi radius are accepted. Ones outside them are smoothly attenuated to 0. And then we have the Fermi width, which dictates how quickly we have that drop off. And as we've seen in our class before, there's an inverse relationship between the Cartesian space and we have Fourier space. So a larger Fermi width leads to a more narrow cutoff, a much quicker cutoff. Okay, next slide, please. So how do we combat this? How do we combat this is that we originally, Young and Lee in 2006 were trying to identify how can they take multi-site studies, in this case genomic data, in order to eliminate the variance between the two sites. And what they came up with this idea is called combat. The original problem could be seen here on the data where they were just trying to analyze the expression of different DNA molecules to nitric oxide. And their clustering algorithm happened to cluster groups, not by treatment or not by time, but by the batches they came in. And that was a problem that was being seen throughout multiple sciences. So in order to combat that, they decided to build a harmonization function that helps reduce the variability between different studies. But the key parts of this is that they wanted both a function that maintained the biological data that was relevant, as well as increase the actual strength of those correlations. So in our paper, that was the motivation for this by Yoon and Lee in 2018, they were looking at the negative correlation between brain connectivity and age. And as we can see here in the pre-combat data, that we have a p-value of 0.06. After we apply combat, p-value is down to less than 0.01. And in terms of the correlation, we see the correlation coefficient gets stronger between whether we're using one form of metric to measure association or another. Next slide, please. So how does combat work? To not get into the deep weeds of it, it's an empirical Bayes estimation. So an empirical Bayes is based on the notion of, if we're looking at these two averages here, 5 out of 10 or 295 out of 1,000, which one tells us more information? Hopefully you guys are going to agree that 295 out of 1,000, there's a lot more trials, we feel a lot more confident in that. So what an empirical Bayes estimation does is it takes into account and has the ability to change very small sample size, which is what we see in FMRI studies, especially on multi-sites. So what it does is it takes a prior knowledge, it samples the entire solution set, it finds what it's the average of the solution set, and it uses that to modulate both of these values in order to find what it predicts to be the actual averages these represent. And in order for this to work, you have some certain assumptions, which are the data has to be similar. You think the data is supposed to look roughly like each other, aren't supposed to be extreme outliers. And as we can see in this graph, these are all the data points before harmonization. And as you can see, they all get pushed in together. It has a great ability to deal with outliers, especially in small sample sets. And then normalization is that we have to normalize the signal for differential output and expression, whether it be genomic data, FMRI signaling, so on and so forth. And the combat code parameters are pretty simple. You have your data, which is a matrix where the rows are the features, the columns are the different samples. You have your batch, which identifies, and you give the ID of the specific site scanner or signal that you want to correct for. Now, this should be the smallest unit of measurement that you believe introduces negative variance into your study. And the mod is a simple parameter that we pass to if there's a certain biological or signal covariate that we don't want to reduce, whether in the case we're talking about before, age or brain connectivity, we simply tell the program, don't touch those variables, look at everything else and try to reduce that. Next slide.
All right, so what materials did we analyze in this project here? We found a study that tasks patients with a simple two-choice reaction task. And essentially, it's very simple. If there is a rightwards arrow, then the participants would press the right arrow. And then if a leftwards arrow was displayed, then they would press the left one. As we can see, there were two different scanners that were used for this experiment here. And they actually have different parameters. These are the anatomical image acquisition parameters. And we can see that there's differences in repetition time, as well as echo time, which can affect the contrast between the gray and white matter, as we've seen. So these are definitely some things that need to be accounted for, and that's why we're using combat to combine the two data sets. So here's an overview of the FMRI data analysis pipeline that we followed. So first we upload or download the raw data sets from OpenNeuro, and then we're using a functional connectivity toolkit called CON, which is an implementation of SPM in Matlab. And essentially, it was developed by a team at MIT for basically functional connectivity pre-processing and data analysis. After the files are then processed in CON, we use combat to batch harmonize all of the MRI files. And then after these harmonized files are outputted by Matlab, we can go ahead and reharmonize, or we can go ahead and denoise them in CON for further statistical analysis. So here's an overview of the pre-processing analysis script here. So on the right side, we can see the raw anatomical images, and then on the bottom, we have post-pre-processed and also harmonized images. And we can see that there's a lot less variance. We can see that previously you could tell that the top five were a little bit darker, and that was due to being from scanner 1. And then the other 5 are from scanner 2. And after the harmonization procedure, they're a little bit, the site effects are accounted for slightly here. And then the actual pre-processing pipeline is a little bit more interesting. So in order to process the different functional scans together as a group, you have to normalize them to normalize the images to M&I space. And then also, since it's a 4-dimensional time course measurement, you have to adjust for motion correction and spatial registration. And then also, during the pre-processing steps, the segmentation of the different regions of interest, such as the gray and white matter and the cerebral spinal fluid, is performed. And that's for later on, it'll be used in denoising and the regression analysis. So notably here, we have one smoothing step, which involves the spatial convolution with the Gaussian kernel, and I'm going to mention that on the next slide here. Okay, so the initial denoising is done by that step in the pre-processing, and it actually results in a signal-to-noise increase at the cost of spatial resolution. So if we can see, the raw functional scan here is a little bit clearer, but there's actually more noise involved. And then once the smoothing procedure is performed, then we see a smoother pre-processed scan, but it is more blurry. And then also the primary denoising is then performed by assigning the confounding regressors, which basically attribute certain degrees of noise of the gray matter, white matter, and CSF to their respective sources of noise. And as you can see, the distribution of the connectivity values actually becomes more normal and approaches 0. And that's because initially we have this skewed right or skewed positive set of connectivity values, and that's generally due to functional, or generally due to patient movement during the functional scan. On the bottom here, we see it's called a carpet plot. It's basically a 2-dimensional representation of 3-dimensional noise for each subject. As you can see, after denoising, the noise tends to go away. So these are a lot of important steps that need to be taken in order to make sure that fMRI data is accurate and usable. And after this, Khan lets you go ahead and do results. You can choose between C to voxel, voxel to voxel, and region of interest to region of interest results. There's honestly so many different results that Khan allows you to output. Here's just a couple of examples here. So I'm not going to get too in-depth into that, and that because I wanted to focus on the analysis portion a little bit more during our project. So that's going to be it from our group here. Thanks again to Professor Liu, Professor McVeigh, and the TAs. And at this time, we would be happy to take any questions.
Great. Any questions from the audience? Yes, there was a question.
Actually, I'm wondering, it's a probability or for that here, it being like it has to be made whatever the probability distribution from like incomplete. So like with like a incomplete data. You're saying would this work with an incomplete data set? Not for any artifact, like estimate any like latent or whatever the standard. No, so the point of combat is to take a small sample analysis for multiple sites and try to eliminate the variance just due to pure site-to-site variation between them. So what it does is tries to normalize the data based off what the empirical Bayes method, which once again says, if I look at the entire data set, I get an idea of what it should be, and therefore I adjust each individual data point accordingly to what I think is a more accurate representation without the noise. Does that kind of answer the question?
Yeah, just an add-on to that. Since most experiments only use one scanner, in those cases where you only have one scanner of data, you don't have any sort of site effect differences, so combat would be irrelevant in those cases.
So I had a question. It wasn't clear to me where was combat used to the fMRI data?
Yeah, so
very end or
there's multiple.
So combat is actually has to be performed on pre-processed data, and that the reason is because this data matrix here needs to have a constant column number. So if we take, for example, MRI files that are of different sizes, we have to first pre-process them and normalize them so that they have the same number of voxels, and that way we can actually enter them into combat. So it's done after the pre-processing step and before the denoising step.
Oh, okay, great, thank you.
Mhm.
Let's move on. So the next group is going to be telling us about deep learning. So is this in person, online? Where are you?
This is online, yes.
Okay, great, go for it.
Awesome. Hello, everyone. Our project is on deep learning for improved MRI reconstruction. Our group members are Jason, Jonathan, Chavi, and myself, Zaid. So for some background, MRI reconstruction is like a time-consuming procedure because we need to sample the case space. So in clinical settings where time can be the deciding factor for a patient's outcome, it becomes like imperative to avoid the need to re-image patients due to artifacts caused by, for example, like the patient's motion and so forth. So our project kind of seeks to experiment with the ability of a deep learning model called an autoencoder to reconstruct MRI images from those with artifacts in them. Because this ability to do so would mean you would spend less time in the clinic re-imaging patients. And so for our project, we use MRI images we found from an open source dataset on Kaggle. And so for some backup, what an autoencoder is, which will be on the next slide. So the image on the slide is like an outline of how autoencoders work, and I'll talk about that in a moment. So an autoencoder kind of consists of two primary steps. The first one is known as the encoding step, and the next one is the decoding step. The encoding step, which in this image is shown in green, it's a function that will map an input. In our case, it will be MRI images with artifacts to a lower dimensional subspace. The second step is a decoding procedure where the lower dimensional subspace will be kind of like decoded to a targeted like image space. For us, this image space is going to be the MRI image without the artifact. So taking a close look at this picture, on the very left column in green, there is this like label called original input. These are going to be our clean MRIs, so MRIs with no artifacts. And then the column next to it, labeled as the partially destroyed input. These are our MRIs that have artifacts in them. These MRIs, the ones with the artifacts, will be fed into the encoder, where they will be mapped to this like lower dimensional subspace shown in red, labeled Z. This lower dimensional subspace does act as a bottleneck in autoencoders because as you bring the input down to lower dimensional subspace, you're undergoing a lossy compression process, a process that will result in loss of data. And this like kind of bottleneck like space can take on like any dimensions, can be a single number. So there's a fair bit of loss. But the decoder will attempt to project this lower dimensional subspace to our desired image space. In this picture in blue is labeled as a reconstructed input. This is our MRI image with no artifacts, hopefully. And towards the end, there's like these like gray dashed arrows on the top of the image, in which you're sort of comparing the reconstructed input to the original input. So we're attempting to get as close as we can to the artifact-free MRI images. And so now I'll pass this to Chavi, who will talk about the types of data fed to the autoencoder.
So we utilize 79 clean MRI images from Kaggle, and to these 79 images, we added 100 different spike artifacts, generating 7,900 spike images in total. And in the table here, we can see the different categories of images and the different distributions. The training images are the ones we use to train our model. The testing images are the ones we use to test our encoder. And the validation images are the ones we use to validate that our encoder was actually performing well. And these are the images that the model never saw during the training process. Here we can see the different distributions in terms of numerical numbers, summing up to a total of 7,900 images. And we have also provided the percent distribution for the different categories, summing up to a total of 100%. In terms of the images we're going to see for our results, they are based on the ground truth image that we see on the left. we are also going to provide the spikes artifacts added in our results, which is the image we see on the right, and as well as the reconstruction image that the autoencoder gave out. And just to emphasize, these spikes are features appearing in an image that would otherwise would not be present in the original object. Thus, this may lead to mismapping of information that would make it harder to provide a correct diagnosis for a patient. In terms of data pre-processing, in other words, what we fed into the autoencoder, we first started with the original image. We then scaled this image to be 256 by 256, as well as removing non-square images. We then took the Fourier transform of these images and added spikes to them. Subsequently, we took the inverse Fourier transform of the image and scaled them to be from 0 to 1. And these are the images we fed into the autoencoder.
So just to discuss the autoencoder a little bit. So this is a simple model we created as a proof of concept just to test out the autoencoder idea. It's a very simple model. There's only 3,217 parameters and just 5 layers. And the decoder layers and the encoder layers are all convolution based. And the main thing that we wanted to focus on with this proof of concept is that each layer has a 256 by 256 dimension with varying dimension of depth. And the 256 by 256 was important to test because we wanted to try not to compress the original image too much between the input and the output. So that was the goal of maintaining that 256 by 256 dimension, was to match as closely as possible the original image resolution. And if we can go to the next slide, we can see how well it did. And unfortunately, it didn't do very well. As you can see, the images actually ended up blurry. In the first column, there's actually more artifacts than there should be. So obviously, this simple model did not perform the way we wanted it to. So we decided to learn from this and create a more complex model, which incorporated some of the learnings from the simple model, which Jason is going to describe next.
So we created a 10-layer, we decided to go for a more traditional autoencoder approach, which will actually reduce the dimensional space into a lower dimensional subspace in the latent layer, and to try to see if it would prove better. So in the first 4 layers, we have the encoder step, which starts off with a 256 by 256 image, goes through convolution and max pooling layers. What max pooling basically does is that it shrinks the dimension down to, as you can see here, a 1/4 of the size, so 128, 128. So this splits up the image into grids of 4, and we'll pick the largest value of each block of 4 and use that as the new value for a picture, therefore like reducing the size by 1/4. More convolution, more max pooling layers until we get to 64 by 64, which is our latent space. Now, the third number, the 256, 256, 64, the 64 basically means we have 64 stacked images on top of each other. And for those of you that are unfamiliar with how autoencoders work, it basically is very, very akin to principal component analysis, where it finds, it maps our original image space into kind of different feature sets. So the 64 is representing that from our original image, it grabbed 64 feature images where it would try to reconstruct the image from. Now, at this max pooling stage, as Zaid and Jonathan mentioned before, we are doing a compression algorithm at the base level, so we are losing a lot of our data. So during our decoder, we have to kind of do these upsampling layers, which will interpolate and bring back, or try to attempt to bring back our image to our original 256 by 256 resolution. Now, this is not perfect in any sense because we are reducing dimensions and interpolating again, so there will be errors with it. But from the simple model we saw before, it didn't really work out. So hopefully this will prove better. And we did train on a lot more parameters. We have over 100,000 of them. And so just for this training set, we only ran it for 3 epochs due to hardware limitations. But as you can see, the loss did go down substantially in just these 3. For reference, this training took about 4 hours to do with over 6,000 data just on our local computers. And so this result is it's not perfect in any sense because we only ran it for 3 epochs, but it already is proving a lot better than the simple model. The reconstructed images look a lot sharper, although not as sharp as the original image, and you can see a lot more details. In addition, there are no more streakings compared to the simple models. The simple models, you can still very faint outlines of streakings, but this one, they're a lot better, and the upsampling did in fact like restore some of the loss of resolution. So just to talk a little bit about what latent space is and what the autoencoder actually does, we have the simple versus the complex model, simple being on the left. So these are the images that the encoder spits out at the latent space. Now the images on the left-hand side are 256 by 256 pixel resolution size. And as you would expect, you see very clear edges. Although the images don't look great, you can still see a lot of definition. You can hardly see any of the actual pixels, and the lines are clearly defined. What this represents is that for each MRI image through the simple model, it tries to map to each one of these 6 images. It tries to reconstruct your original image through these 6. And then on the right-hand side, you have the more complex model, and I think from the scale, it's only 64 by 64 images. Now, these images do look a lot better, but you do see the loss of resolution. You can see individual pixels a lot more clearly than on the right-hand side, and the edges are more blurred and less well-defined. In the more complex model, we only display 6 due to like size restraints, but there are 64. There should be 64 of these images out there, and they're all just very similar to this. So we are reconstructing on a lot larger feature space. And so Saeed will go and talk about our conclusion a little bit.
All right, so in this project, we saw a few things. First, like a deeper, more complex model does give us a better performance. So in the future, we would want to explore this correlation between complexity and like performance and try and figure out like how far we can push this computationally in like a feasible manner. Secondly, we did also see that there's a loss of resolution as we go through the autoencoder because of the lossless step due to the modeling to lower dimensional space. So in the future, it would be like worth trying to find like a model that kind of preserves some of the dimensionality and resolution. And so one such model is the unit autoencoder. An image of that is shown on the slide. It goes through the same steps as a traditional autoencoder. However, if you look at the image, there are these like gray dashed lines between the earlier like kind of like layers towards the later layers, like from the input to the output. This kind of like helps concatenate earlier stages to future stages to kind of like help and preserve some dimensionality and resolution. So we would love to repeat this project with this autoencoder if we get a chance. Something else worth exploring is, for example, how would these autoencoders work with something that isn't just spikes? For example, if we have an aliased image, would it still be able to reconstruct the original MRI image from an aliased image? That would also be worth exploring. So overall, we'd like to do all this. Any kind of progress in like this area would like help reduce like patient time in like MRI imaging. Yeah, so thank you so much for listening. We have citation on our next slides, and I will take any questions you may have right now. Yeah, thank you.
Great. Any questions from the audience? Yes, question.
So when you are doing your autoencoder, what kind of loss function?
The question is, what kind of loss function are you using for your autoencoder?
So for the loss function, we use a binary cross entropy for the more complex model, and for the simple model, we use just mean square.
Question is, why not use KL divergence?
For these, when we ran the models, we looked at, we just saw that the classic one that they use for images, for MRI images, most people use the cross entropy loss function. We did try to experiment with L1 norm also, but that just didn't do anything for our model. It just crashed, so, or not crashed, it just gave us like black images.
Okay, great. Any other questions? Yes, question. could- So is the question? Oh, okay. The training and test data set split. Was that, did you add the spike for all of them, or what's the question? Oh, right. Okay. So the question is, was the training, I guess I was understanding, was the split between training testing done on the 7,900 spiked images, in which case there could have been some of the validation set data was already seen with another spike. So when did you split it? Did you split all the original images first and then add the spikes, or did you add the spikes and then split the data?
We split the data. So this is where we got a little sloppy. When we added the spikes, we didn't shuffle anything. So there was 100 of patient 1, 100 patient 2, all the way down to 100 of patient 79. And then when we split the training data, we chunked the training and testing images. We try to keep with the 75-25 split of training images to like testing validation set. So there is some overlap between the images that were between the training and testing images, and that's why we have the validation set. So the validation set shouldn't have any that are with the training and testing images.
Okay. There's another question way back there. Yeah, so the question is, so is the question, why didn't you use case-based data as the input to the autoencoder, or what? Yeah, that's my understanding. So the question, I guess, would be, did you guys consider using the case-based data as the input?
Yeah, we tried running the case-based data through the input, but then the problem with that is like case-based data deals with, if we want to encode and capture the entire case-based data, it's in complex form. Mm-hmm. And if we take the absolute value and run that through, we can't get a reconstruction because it's losing half the data. We did try running the absolute value of the case-based through it, so just magnitude, but we just got black screens every single time.
Is there a follow-up question? Okay, that's good. Yeah. Yeah. So can the autoencoder handle complex data, or is that a limitation of the autoencoder?
From what I understand from autoencoders, that if we want to have it take in complex data, it's a completely different network that's a lot more complicated to implement.
Okay, thank you. Thanks, great talk. Okay, why don't we do one more talk before we take a break? So we have a group doing compressed sensing. Is that in person, remote?
No, we're all online.
All right, go for it, Ben.
Okay, hello everyone. Today we are talking about compressed sensing, and I'm presenting with Tianyang.
Speak up a little bit.
Sorry?
Could you just speak up a little? Just
okay, sure.
Speak more loudly, just a little bit.
Okay, sorry. Hello everyone. Today we are talking about compressed sensing, and I'm present with Tianyang Xu, Yuanshan Wu. And firstly, I would like to briefly introduce MRI again. Conventional MRI techniques are designed to use the well-known Nyquist-Shannon theorem. It says the digital sampling of the MR imaging must be performed at least twice as rapidly as the highest frequency expected. To obtain a high spatial resolution imaging with a high signal-to-noise ratio, the imaging should be acquired with a lot more case-based data. This, however, will increase the acquisition time and therefore increase motion artifacts. So how to reduce the acquisition time of MRI while maintaining imaging quality is a great challenging. Compressed sensing is an extension of Nyquist-Shannon theory that only a small number of measurements is enough to recover an imaging with good quality. It is an acceleration techniques allows for faster MR imaging since it acquires and stores less data, therefore operates more economically. In order to apply this specific imaging application for MRI, the unknown signal should be sparse. Sparsity refers to a signal that most of these coefficients are zero and very few non-zero. Most MR imagings are not sparse, but can transform into some sparse domain, like the wavelet domain. And this transform sparsity determines the compressibility of the images. And the imaging on the right shows the non-sparse and sparse representation for the 1D and 2D signals. In order to find the minimum of coefficients and promote sparsity, L1 norm is performed. Some coefficients are suppressed with L1 norm because many small coefficients tend to carry a larger penalty than a few large coefficients. And an iterative thresholding algorithm is applied to solve L1 optimization for the compressed sensing. Another key point of the compressed sensing is the incoherence of the signals. In other words, K-space is undersampled. Incoherent undersampling is important because it can avoid aliasing artifacts. However, uniform random undersampling is not practical. It does not consider the energy distribution in K-space, which is far from uniform. Most of the energy is concentrated close to the center of K-space and rapidly decay towards the edge of the K-space. So in general, undersampling patterns usually follow the exist trajectories, such as the radical or spiral. As you can see the image here, from left to right on top row are the undersampling patterns of the radical, spiral, and truly random sample in K-space. The second row are the corresponding reconstructed imaging. As you can notice, the truly random sampling has the worst result. The last step of the compressed sensing is the reconstruction. The imaging must be reconstructed by a nonlinear method, which enforces both sparsity of the imaging and the consistency of the reconstructed data. In general, original imaging can be reconstructed with sparse optimization algorithm. If this reconstruction is exact, the imaging is said to compress with no loss. In other words, the differences between the original and reconstructed imaging is zero. If the compression is lossy, it results in loss of some information, but is not typically not perceivable to the human eyes. And this lost information is the coefficients close to the zeros and is not used for reconstructions.
So our model starts with generating an undersampling data acquisition from the original image. We control such undersampling ratio by generating a probability density function with our main probability defined as a ratio of undersampling. Like in figure A here, we use a multivariate probability density function with round shape and generate the probability density function at a mean probability of 0.333. So that we assume 1-to-3 ratio of undersampling. Down there is a square PDF generated from another toolbox. In the figure B, using these probability density functions, we can randomize and generate an undersampling mask, which can be used to undersample the K-space slice of our 3D objects. The result K-spaces are showing.
Very noisy. Is there a microphone you can use, or?
I'm using my microphone right now.
Okay, maybe is it next to your body or something, or yeah.
Oh, okay.
Okay.
Is it better now?
Yeah, that's better. Thank you.
Shall I repeat or just continue? Sorry.
Just continue. Thank you.
Oh, okay. The resulting K-spaces are shown in figure C with the inverse transformed undersampled image. We will talk about the differences compressed sensing results brought by undersampling strategies. But for here, these two type of undersampling mask did not show significant differences in the results. After obtaining such an undersampled acquisition, we applied wavelet transform to the image. We applied in the 6 levels discrete wavelet transformation with Dabecki's wavelet length at 4. The demonstration of the wavelet transformation started with figure A. The undersampled image is filtered with a periodic convolution with a wavelet at high or low pass filter along the x-axis and downsampled to half of its size. After that, the result of 2 images are treated with y-axis periodic convolution with wavelet and split based on their frequencies along the half-size downsampling. The results are shown in figure B, where the LL means both low pass filter on the X and Y axis. Therefore, the image still maintains most approximation of the overall image information. The HL has high pass filter on the X axis, so you can still see the vertical details of the image. LH has edges on the horizontal way, while HH has only diagonal features of the object. Coming to figure C, we tried DB12, which has longer length than DB4. The wavelet. Therefore, the periodic convolution is more smoother, and you can see more edges and features in the high pass filter area. Hard filter is a special case that is the discontinued wavelet, and also we examined this filter wavelet in our Matlab code. The transformation with the wavelet also means now we can go into an iterative process to finalize the sparsity details in the undersampled image. We put a soft threshold on the wavelet transformation shown in figure A. After that, we perform the inverse wavelet transformation. In figure B, start with the image LL. It's convoluted with the wavelet at inverse time modulation, and the result is resized to twice of its length. Then its corresponding high pass image located right beneath it. It has a similar operation and it is adding back to the image. The operation then repeats on the x-axis. So now we have a level 1 reconstructed image. The operation continues until it goes all levels and back to 512 pixels in length, which is our original image size. The result is then used to the inverse undersampling filter to retain the details that the undersampled image lost. And then it is added back to the undersampled image. By doing this, an L1 norm is complete, but to minimize that norm, we are doing iterative cycles of it. So figure C shows a 30 steps iteration process, and the outcome is actually pretty good after the 10 cycles. To sum up our model approach, here's a mathematical illustration of the image reconstruction process. Equation A is the minimizing problem we need to solve in the compressed sensing MRI process. It has 3 parts. The L2 norm is solving the least square feet of reconstruction to the original image. And also it has a total variance of the reconstruction image. And the L1 norm of the wavelet sparsity regulation factors. We focus on the wavelet sparsity regulation part, which is equation B. So we apply this iterative method with a discrete periodic convolution with a wavelet function and treating the undersampled image by iterations of cycles.
So then we run our simulation. In our simulation, we loaded the MRI 3D axial images from Matlab imaging processing toolbox. And there are 27 slices of a human cranium. Firstly, we want to test our idea of different undersampling mask. So firstly, we apply 2 different methods. The images on the top is the complete random samplings. And the first image on the top is the mask of the complete random sampling. And they are all threefold undersamplings. And the image on the right, the first image is the fully sampled images. And the middle one is the linear reconstructed images. And the last images is the compressed sensing with 50 iterations. And the image on the bottoms are variable density random undersamplings. The complete random undersampling introduces noises that cannot be reconstructed properly due to, we think, 2 reasons. The first reason is that most energy of MRI images located in the center of K-space. And also we still want to fully sample the readout directions so that we can only undersample on the phase encoding directions. Another really important point we have already mentioned is the natural biological images are not sparse in pixel representation. And to remind you, the sparsity here means images have few significant pixels with non-zero values. So we perform the wavelet transformation by using the DB filters, which generate a relatively more sparse images compared to other filters, such as Haar filters. So the workload for us is to Fourier transform the image to K-space and then undersample it with variable density function. Then we transform the undersampled images to wavelet domain with utilizing the soft thresholding method to reconstruct. The equation on the right is a mathematical representation of soft thresholding, and the U here is a pixel value in wavelet domain, and the lambda is the threshold values. If the absolute value is smaller than the threshold, the signal will be truncated to 0. Otherwise, the value will be scaled down by the percentage difference compared to the threshold values. We test out several different threshold values, and we chose the 0.025, which can help us improve the image quality the most. So usually the lambda value is smaller than the noise levels. The movie here shows the fully sampled images, linear reconstructed images, and compression sensing with 50 iterations. And we believe all the slices improve their qualities. So in order to show the improvements, we took one of the frames and also the zoom in view of our results. Clearly, the small structures and edges are blurry for linear reconstruction, like here. And by 50 iterations of compressive sensing, the edges are sharpened, and the small features we can also observe. In the end, we're gonna discuss the advantages and the drawbacks of compressed sensing. So compressed sensing can reduce scan time and improve the image resolution. And it can also play an important role in many applications that are limited by scan time. For example, infants are harder to keep still in MRI machines, which will cause blurry images. By reducing the acquisition time, it could avoid these problems. And the drawbacks of our method is that compressive sensing reconstruction algorithm usually is computationally intensive. For our 3D images, we took 50 iterations for each scan, and there are 27 scans. So that in total 1,000 iterations. It could take like 3 to 5 minutes. In order to solve that, we could probably use parallel computation. And also in certain applications, the artifacts may appear due to the small high frequency oscillation in wavelet-based reconstruction. And here is our reference. Thank you so much.
Great, thank you. Any questions from the audience? Okay, I had a question. So how did you do your linear reconstruction? I mean, they didn't look terrible. I mean, so how did you do that linear reconstruction?
Yeah, I think the way we did it is we inverse. I think we just perform the inverse Fourier transform, and then like we applied like certain thresholding, and then we do the Fourier transform back. Yeah.
So okay, so you basically so you undersampled, you just sort of randomly undersampled in K-space and just did inverse Fourier transform on that, and then thresholded in the image domain.
Yes, and also for this slide, when we construct this image, the undersample ratio is 1:3, and by using that incoherence K-space undersampling method, the most of information is actually can be still preserved for the image.
Okay, so yeah, I'm just surprised there's not more artifacts there.
Yes, because we didn't fully randomize that undersampling. We still have the probability density function to control it from behind.
Oh, okay, so this is with the density, sort of higher density sampling in the middle. Okay.
Yeah, it's weighted. Yeah.
I see. Okay, great. Any other questions? Okay, well, I think this is probably a good time for a break. So why don't we do a 5-minute break? We'll start the next talk at 1:15. So it's 1:10 now, so 5-minute break, and we'll resume. Okay.
MRI room may not get the medical attention that they need, but with portable MRI, the medical attention can be move to them. Lastly, portable MRI uses nonlinear phasers to reconstruct images that improve the overall image quality and parallel imaging performance, which will be looked at in closer detail in the following slides.
And for the next 4 slides, I'll be talking about some MATLAB work we did to show how portable MRI could work in a computational sense. This first slide summarizes how traditional MRI physics uses efficient imaging, and by efficient, I mean computationally perhaps not as intensive as portable MRI physics. As we know, there are really 3 criteria you need to fulfill before you reconstruct the inverse Fourier transform. You need to create the spatially varying phasor pattern, then multiply the object by the phasor pattern to obtain the product. And finally, you need to integrate the product all over space to obtain the Fourier transform. And then from there, you can use the inverse Fourier transform command built into MATLAB to reconstruct an image. So here you see an example of what we've done somewhat in our homework, where we utilize a linear phasor, and from there we compute the Fourier transform values. We can use the IFFT2 command, which is the inverse fast Fourier transform built into MATLAB, to reconstruct the image at a very good and convenient pace. But when we move to portable MRI, that is not so much the case. Fundamentally, portable MRI uses nonlinear phasors. And this can be problematic in the computational realm because, as you can see here, when you use the same exact code that was used in the previous slide and you look at the end result, you produce something very distorted and something unlike the original object. And notice that not much has really changed. Here, we are just simply squaring the kx and ky terms to create a nonlinear gradient. So just a subtle difference can really slow down and really alter the effects of the overall result of the Fourier transform. Yeah, here's one solution that you can do. A solution that can be used is nonlinear reconstruction via vectorizing the object. This was something that was done in our homework 6, I think, where we perform image reconstruction, but not using MATLAB built-in features like IFFT2, but instead using a method of vectorizing the object and using the pseudo-inverse. So as you can see here, we are considering a different object because of our lack of computational power to vectorize the sagittal head image. But as you can see, that when we get the pseudo-inverse and we use the pseudo-inverse, we use the method of vectorizing the object, we can get a result that is somewhat similar to the original object, but not quite the same. The last type of solution is using nonlinear gradients in a hybrid signal reconstruction form. In this method, we can compute the Fourier transforms of kx and ky independently, as outlined by the code there on the right, giving opportunity to use built-in MATLAB functions to reconstruct the image if linearities or nonlinearities are present in one of the 2 variables, kx, ky. In this example, we have a linear phasor, but wanted to illustrate how this would be performed computationally and how it can be just as effective as other reconstruction methods, such as built-in MATLAB features and vectorizing the object.
So how does portable MRI works, right? Unlike the traditional MRI system, which aim for a full body scan, portable MRI mainly focuses on imaging a specific part of the body. There are many groups are trying to make the MRI system smaller and lighter. Some of the most popular directions are reducing the weight and size of the magnet, an integrated magnetic grading coil, RF subsystem, a better console, and computational systems that can shift the burden from the hardware to the software side. In this presentation, we focus on the magnet specifically, and we have organized a list of papers that was able to successfully reduce the magnet size and weight. However, I will only be able to introduce a portion of the paper due to the time limitation, and hope it will give you a taste on how portable MRI works. The paper we picked is from Lawrence Wei's group, where they use the Hall-Batch permanent magnet array for this specific design. So what is a Hall-Batch array, right? The simplest way to explain this is if we arrange the magnets into a ring shape, and for each magnet oriented them with a very specific direction, the magnetic field inside of the ring will be uniform and have no field outside of the ring. By this property, the magnetic energy is contributed by the permanent magnets, which does not require a cooling system, shielding, or current source. So let's take a closer look into their design. The pictures on the top shows how the device looks like, and the device is only focused on imaging the brain. In this design, instead of using a superconducting magnet, they used the N42 and N52 graded neodymium permanent magnet with the Hallback array, of course, which was showed on the picture at the bottom of the slide. However, a highly homogeneous Hallback array with this level of size and shape constraints is super difficult to achieve. So instead of focusing on working out a highly homogeneous polarizing field, a magnetic field variation is shifted into a built-in field gradient for the imaging encoding. This allows a reduction in the magnet size. It eliminates the need for a radial gradient in the traditional electromagnetic system, reduce the acoustic noise, power and cooling system are no longer needed, a safer, lower cost, and less footprint design can be achieved. The system can create overall a 0.08 Tesla of magnetic field, and the overall weight of this system was only 230 kilograms.
So there are a few portable MRI designs and concepts that were built by researchers that only image certain parts of the body. The first one on the left shows a development of a small car-mounted MRI system for human elbows using a 0.2 Tesla permanent magnets. The graphic under it shows the detection of rheumatoid arthritis bone erosions by two different dedicated MRI units and conventional radiography. And the graphic on the right shows some wearable technology which is shown as a knee brace for imaging the knee and below the waist. In terms of clinical applications, there's only one company that is FDA approved for their portable MRI technology, which is known as Hyperfine. Their technology allows to bring portable MRI to the patient, and their main use is for detecting strokes in humans. The device is designed for imaging the brain and head region, and you can plug it into a normal 220-watt electrical outlet in hospitals or designated remote regions. It uses a wireless tablet to set up the device, monitor the patient, and review the reconstructed images. And just like Audrey mentioned before, it has very low magnetic field, 64 milliteslas versus a traditional MRI of 1.5 Tesla magnets. The concept of portable MRI has a very strong and huge potential to change the medical imaging diagnosis. Hyperfine's portable MRI can currently image the head and brain region, but they should be working on methods for imaging other parts of the body, just like a normal MRI does. A portable MRI allows for rapid imaging of patients with low labor-intensive movements. This is an advantage for fatal patients in the ICU who are on ventilators and life-sustaining devices. It also allows health professionals to learn important information bedside with their patient. And lastly, it is, if you were to increase the magnetic field, you can provide a better imaging modality. Portable MRI is limited by imaging specific parts of the body. And so if they're able to image the entire body at once, they would provide the next step in this technology. Those are our references, and thank you so much for listening to our presentation.
Great, thank
Any
you.
questions?
Great, any questions from the audience? Okay, let me ask a question. So when you're talking about that, the Hallback design, you mentioned there was pre-polarized as well. So are they using both a permanent magnet and a pre-polarized coil? So the next slide. Yeah, so is there a pre-polarized B0 system in there as well, or is it all the permanent magnet?
So I think there are many different methods. So for this specific one, the permanent magnet with the Hallback array does have a pre-polarized field here. And I also believe they have, if I didn't remember wrong, they also use something else there to create the polarized field here. Yeah.
Okay, great. Yeah, so it sounds like they probably have an additional coil there where they pulse the field intermittently to do what's called pre-polarization, and they turn it off.
Yeah.
Okay, great. Any other questions? Okay, well, why don't we, thanks again, and why don't we move on to the next talk, which is another compressed sensing talk. So is the group in here? Okay, come on up.
Hello? All right. All right, so our topic is on compressed sensing, and my name is Don. This is Carol, and this is Lena. So the idea of compressed sensing is originated from the idea of Nyquist sampling theorem, which we'll be mentioning a little bit later in our slides. And compressed sensing is signal processing technique for efficiently acquiring and reconstructing a signal, finding solutions in underdetermined linear system. So in other words, the compressed sensing theory permits linear projection of a high dimensional signal into a lower dimension, and later allowing recovery of a signal from the projections. And for applications of compressed sensing can be used in many fields like, of course, medical imaging and mobile camera sensor and facial recognition applications. So the key idea behind this conventional compressed sensing method is to extract and retaining only a small number of high energy transform coefficient and encode this while discarding the remaining ones. Why we're using compressed sensing? Because of conventional sampling followed by compression, the sampling and acquisition stage deal with a lot of data and require efficient sensor and storage capacity. So which will result in longer scanning time and increasing in the cost. So to solve such problem, we exploit low dimensional structure to acquire signals. Nyquist-Shannon's signal sampling theorem optimizing the sampling processing by suggesting that if a system uniformly samples an analog signal at a rate that exceeds the signal's highest frequency by at least a factor of 2, the original analog signal can be perfectly recovered from the discrete value produced by sampling. However, in many complex applications, the resulting Nyquist rate is so high that it ended up with far too many samples that we expected. Then we use compressed sensing to optimize the process of sampling and compressing that rather than first sampling at a high rate and then compressing that sample data. We would like to find ways to directly sense the data in compressed form, reconstruct the image with this data of a series of linear or nonlinear algorithms. So we make compressed sensing. So what makes compressed sensing possible in MRI? Because of the freedom with that, we're able to choose the sampling trajectory, play a major role in making compressed sensing possible in MRI. Relating to our course material about the gradient, the trajectory Kx and Ky, this waveforms along with the associated RF pulse used to produce the magnetization. We call pulse sequence. The integral of gradient waveforms trace out the trajectory in a spatial frequency domain, which is what we've been working on with a lot this quarter in a K-space. So we transform the image to K-space in order to compress sensing to work better. Because for one requirement for compressed sensing is the sparsity of image. As we were mentioning for a transform, so we transform the image to K-space, and then after we ran the image through the compressed sensing, then we have to inverse Fourier transform back to have the image in the image domain to look at the picture. So these are the two images that we worked on. This is the Sharp-Logan phantom, and on the left is the Sharp-Logan phantom in K-space. So there are 2 points on why we need to transform it into frequency domain, because MR system encodes spatial information and the sparsity transform of MR image. So image content can be stored as a vector of sparse coefficient and then encodes the sparse vector by approximating the most significant coefficients. and ignoring the smaller ones. Sampling linear combinations or sampling individual Fourier coefficient. So compressed sensing is claimed to be able to make accurate reconstruction from a smaller subset of K-space, and rather than on an entire K-space grid.
Okay, so then we're gonna talk about the compressed sensing. And generally, there are two key components involved in compressed sensing, which is the compression and reconstruction. So I want to address a little bit on compression part first. So compression means to reduce the data volume and achieve a lower rate in the digital representation of radiologic image with minimum loss of image quality. In here, we have the input X and random matrix phi. Sorry. Matrix. With these two factors, we can generate sparse vector and compress it to a lower dimensional vector signal Y for the further reconstruction. So the first step before compression is sparse transform, which is here X is one column from M times N matrix. And we're gonna only take one column, for example. So within the process of sampling, X can be written as a linear combination of alpha and psi, where alpha is the coefficient of X on the base of psi, and psi represent the sparsely transformed matrix, which will ensure that it's gonna preserve the information of the signal X. So after the image run transform over the base of psi, since most of the location in the spatial domain of image have small coefficients, they're gonna be dropped off and counted as 0 in alpha. So there are only S times alpha j equals non-zero. And the notation here just show that there will be only S number of L sub 0 norm of alpha counts non-zero. And this represents the information we preserve after sparse transform. And in this process, there one thing we have to make sure that the S, which is the number of coefficient kept in alpha, should be much more smaller than the size of input signal. So after we drop off the small coefficient signal over sparse transform, the next step is how we create a low dimensional structure to exploit less data than original image, which is the step of compression. So here's the figure 11 is the process of compression. And the basic idea of the compression related to the underdetermined system. So generally, if we have the same number of square matrix and image, we're gonna come up with the same number of measurements. However, within the underdetermined system, the number of unknown factor is more than the number of equations, which means that we'll have more than one solutions and it makes our problem ill-posed. So how to find our optimal solutions? So in the process of compression, since small vector Y can also be written as a linear combination of compressive matrix phi and sparse vector alpha. And since our signal alpha is sparse or compressible, which limits that the compressed signal Y only depends on the small number of degrees of freedom. And we usually pick the minimum norm solution. And also we have to take the reconstruction efficacy into consideration. So here is the overall idea of the compression. So consequently, much smaller vector signal Y with M samples generated from N sample signal vector X via multiple matrix. The compressive matrix psi with the and discrete Fourier transform matrix phi, which the size is N times N. So this is the downsampling image we generated with our code, and we can see many data has been dropped off after compression. And the figure 13 is the inverse Fourier transform image, and figure 14 is how they look in the K-space.
So after downsampling, we successfully transform original signal X into a much lower dimensions Y. Then the next problem is how to reconstruct the measurement vector Y to X delta through the matrix A. We used an algorithm called orthogonal matching pursuit. So based on this method, the column of A is a set of bases of normalized 2D discrete cosine transform matrix, which we say DCT. And DCT is often used in image compressed, DCT is often used in image compression applications because most of the visual significant information about the image can be concentrated in just a few coefficients of the DCT. So then we find out the contribution of each basis to Y. We call it the weight of the corresponding basis. Later, we use the Y to subtract the weight times its corresponding basis to get Y prime and find the contribution of left basis on Y prime. So we keep repeating this process until the Y prime is small enough. The picture in the middle is the reconstructed K-space image, and we did the inverse Fourier transform to get the reconstructed MRI image. So as you can see, the reconstructed image still have a lot of noise in there. So some pre-processed methods should apply on it, such as the soft threshold or total variation. The total variation should be applied to denoise our images. So in conclusion, compressed sensing provides a new approach to reconstruct image that's similar to original image with time saving, for it accelerated the process of MRI by downsampling the original image. Besides, it reduced the data we need to store as well. However, one of the biggest problem is the compressed sensing is that it takes too long time during the reconstruction process. And the reconstructed process also used a lot of memory. So currently researchers will combine the compressed sensing with machine learning or deep learning to reduce the reconstruction time and make it a more efficient technique. And this is an example of to support that compressed sensing is time saving. Here is a series of images of lower leg muscles, and A is the image with the full data. B, C, and D are the images accelerated by the factor of 3.65, 4.94, and 6.42, which means that there are many data will be dropped off during the acceleration to verifying degrees. But when we look at the inverse Fourier transform image, there is very no difference by eye, and the discrete valid transform also looks very similar. Is there any question? Yeah.
Thank you. Any questions either from, yes, question back.
I have a question. Did you guys find out the ratio of the WK of that one single pixel? Go back to 2 slides and compress image.
Yeah, I think I used the pixel 1 for the reconstructed image. Yeah.
Other questions? I was a little confused. The compressed sensing was you took your K-space data, you undersampled it, and then you used the discrete cosine transform to reconstruct an image space directly, or?
We reconstructed in K-space directly.
Oh, so you're trying to come up with a compressed sensing representation of what the K-space data should look like.
Yeah.
Okay. Why would you the K-space data? What in terms of assuming that it's going to? So you're saying the K-space data should be sparse in discrete cosine transform space. Is that sort of your basic assumption?
Yeah.
Okay. All right. Okay. Thanks. Anything else? Okay, great. Thank you again. Okay, our next talk is on MRI for Alzheimer's disease. Is this in person, online?
It's online.
Yeah, we're all online.
You guys, let's see. So this is going to be online talk? Yes, I guess so.
Yes.
Okay, so hi everyone. Today we will be talking about MRI for Alzheimer's disease diagnosis. So to start, Alzheimer's disease is a type of dementia that affects memory, thinking, and behavior. An estimated 6.2 million Americans aged 65 and older are living with Alzheimer's disease in 2021. Advances in healthcare have increased life expectancy, leading to an increase in the aging population, making it probable that the number of people with Alzheimer's will continue to increase. This will be a huge burden on the healthcare system and a hardship for people living with the disease, making it even more necessary for early detection of the disease to offer early treatments. The exact cause of the disease is unknown, but it is associated with a buildup of proteins in the brain. Alzheimer's can be diagnosed based on clinical changes in cognition, such as memory, problem solving, or language usage. Imaging is also used to help diagnose, but scans alone are currently not enough to diagnose Alzheimer's. Scans can be used to rule out other causes of dementia, such as hemorrhaging or brain tumors, and to see any atrophying in the brain. Alzheimer's is associated with a loss of brain cells, which can lead to shrinkage in the brain, especially in the hippocampus region, where a lot of the atrophying is seen initially in patients.
So a little bit more about the brain. The brain contains white matter, gray matter, and cerebrospinal fluid, or CSF. The white matter is in the deepest part of the brain and it's made up of myelinated axons, while gray matter is in the outer surface of the brain and it's made up of neuronal cell bodies. You can see the white and gray matter in the image at the top right. Communication takes place in the white matter and processing takes place in the gray matter. Neuron loss caused by Alzheimer's disease leads to a reduction in the gray matter volume, which can be visualized on an MRI. CSF is a fluid that surrounds the brain and spinal cord. It cushions and protects the brain while also delivering nutrients and removing waste. These brain components have different relaxation times, T1 and T2, which are important to consider when selecting sequence parameters for an MRI scan. T2 relaxation is the time required for the transverse magnetization to fall to about 37% of its initial value. And T1 relaxation is the time required for the longitudinal magnetization to reach about 63% of its maximum value. Solids tend to have a shorter T2 time because the molecules are relatively immobile, and liquids such as CSF have longer T2 times because the molecules are highly mobile. T1 is shortest when the molecular tumbling rate is equal to the Lamar frequency. Molecules tumbling faster or slower than this frequency have longer T1s. In general, small mobile molecules have higher T1 times, and then as molecular motion slows, like in proteins, gray and white matter, T1 starts to increase again. And then one reason T1 is less for the white matter than the gray matter is because there's more fat in white matter because the axons are myelinated and fat has a shorter T1 time. You can see the average values for T1, T2, and proton density for CSF, white matter, and gray matter for a 3 Tesla scanner in the table on the bottom right. And these are the values that we will use later on in our simulation. So for some more background information, to create contrast in an MRI, the two sequence parameters TR, which is the repetition time, and TE, which is the echo time, can be adjusted. TR is the amount of time between successive pulse sequences, and TE is the time between the RF pulse and the echo signal. And you can see that in the figure at the top right. The most common MRI sequences are T1 weighted and T2 weighted sequences, which are achieved by adjusting these sequence parameters. So T2 weighted images are created by making TR very long as compared to T1, which essentially eliminates the term that contains the T1 value, as you can see in that equation. And this means that T2 properties of the tissue have more influence on the image. Conversely, T1 weighted images are created by making TE very short compared to T2, which essentially eliminates the term, meaning that meaning the contrast and brightness of the image are predominantly determined by the T1 properties of the tissue. So in a T1 weighted image, the CSF is dark, the white matter is light, and the gray matter is gray. And you can see that in the T1 weighted image at the bottom right. And this therefore emphasizes the contrast between gray and white matter. The sequence that we'll consider is T1 weighted, which we'll go more into in future slides.
So the sequence that we are using is called Magnetization Prepared Rapid Gradient Echo, or MP-RAGE. It captures, it is an RF imaging sequence that is one of the most popular sequences for brain imaging. It captures high tissue contrast for brain tissue and provides high spatial resolution with whole brain coverage in a relatively short scan time. As mentioned before, it is T1 weighted, as the TE interval between the pulses is very short. This makes it effective at imaging brain tissue, in particular, as gray and white matter show up in good detail and contrast. MP range is a gradient echo sequence and consists of a large 180-degree inversion pulse followed by a rapid sequence of RF pulses and echoes, which can be seen on the graph. TI is the inversion time, which is the interval between the inversion pulse and the gradient echoes. TD is the delay time, which is the time between the last gradient echo and the next inversion pulse. Both of these features can be seen on the graph as well. These graphs show the intensity levels of different parts of the brain during T1 weighted inversion scans while changing specific parameters for each graph. The leftmost graphs show intensity versus inversion time. So this is the delay between the initial big inversion spike and the echo measurements. As you can see, shorter inversion times result in a greater contrast between white and gray matter on the which is shown on the bottom left. The middle graphs show intensity versus TR. Longer TR times also result in higher contrast. The graphs on the right are from the presentation slides in lectures and show a typical T1 weighted sequence with no inversion. And as you can see, the contrast instead drops off over time. So MRI, MRIs are very good at detecting imaging brain tissue and therefore can be used to detect early stages of degeneration and atrophy due to Alzheimer's. MP range in particular is good at this due to the high contrast and spatial resolution it offers, particularly between white and gray matter. It can also detect the shrinkage and atrophy of gray matter in a patient's brain and can be fine-tuned to detect finer detail depending on the patient. The image shows different inversion times, and as said before, shorter inversion times are showed to provide more detail. Picture A in the top left has the shortest inversion time and the highest detail, whereas D has the longest and least amount of detail.
So as mentioned by Sam, MP range is a gradient echo sequence where 180-degree inversion pulse is followed by rapid RF pulses. The equations above are proportional to the signal intensity at the ith RF readout pulse. Immediately, we can see that if we compare two different tissues, the T1 values will impact the constants gamma, delta, phi, and mu. The constants TD, N, theta, alpha, and tau represent the delay time, the number of pulses, flip angle of the RF pulse, inversion angle, and the spacing of the echoes, respectively. These constants were chosen based on the 2014 research paper on the parameters MP range, which simulated contrast between white matter and gray matter with varying factors. Before we proceed to the simulation of the equation, we want to build a little bit of intuition of what we can expect by examining this mathematical representation. First of all, the constants with the exponential form like gamma are going to be to less than 1 if we consider something like the graph of e to the negative x. Phi is actually just 1 because the TD is considered would be 0 in the simulation. And next up, we see the MEQ, which is the steady state magnetization, will be a constant between any readout RFs from the same tissue. As there's no mention of the iteration number in the equation. Likewise, delta and gamma are the same throughout, so we can conclude that the mu to the ith power term is the primary driver in the difference between the ith and the ith plus 1 readout. If we consider the fact that mu will be less than 1, when we raise it to the ith power, we actually get smaller values. So everything combined, we can reasonably conclude that the signal intensity will decrease as we have more RF readout pulses for the same tissue. For different tissues, we would have to consider the different proton densities in the MEQ equation and the different T1 values. So when we take this all in mind, we did a quick calculation with m not equal to 1 to see how the signal intensity progresses. So before speaking on the simulation results, when I say that the signal intensity of the readouts, I want to clarify it's the signal intensity of the region in the red box on the image on the bottom. So when we refer to the kith or ith readout in the reference, it's in reference to these RF pulses, each one with a 12-degree flip angle. So utilizing the mathematical foundation we built in the previous slide, we can understand a bit of the behavior of the signal intensities as we proceed through the pulses. Looking at any of the tissues, we can see that it's decreasing as the number of increases from the previous explanation that between pulses we're raising value less than 1 to a power, effectively reducing the value as we proceed along the readout pulse. That being said, we can consider how T1 proton densities impact the graph, as the CSF has a density about 1/4 of the white matter and less than 1/2 of the gray matter at the first pulse. Overall, we can see that there's a separation between white and gray matter, which in terms of image quality would be indicative of contrast. We can see the separation decreases as we continue because the rates are mostly based on T1, and we can see where the white matter, which has the lowest T1, decreased at a faster rate compared to the CSF, which had the highest. The simulation was used to visualize the signal intensities along the readout pulse number to see that the contrast between white matter and gray matter is possible using the equations from the previous slide.
So in conclusion, our project has shown that the MP-RAGE sequence provides a good tool for radiologists to differentiate the contrast between white matter and gray matter in the brain scans. This is helpful in the early diagnosis of Alzheimer's, so the rate of progression of the disease can be slowed down. MRI in general has been able to provide the necessary markers for understanding the progression of the disease. Different sequences have been found to produce T1 and T2 weighted images as a way to receive information from both. However, MRI alone is not enough for the optimal diagnosis of Alzheimer's, as there are cases of misclassification due to similarity in brain images. Therefore, the field is currently moving towards incorporating MRI with machine learning, which uses algorithms and statistical models to analyze the MRI data. This approach has been widely proven in different studies to be able to detect patterns in the data in order to differentiate between an Alzheimer's brain and other changes that could be happening in the brain. Which will lead to earlier detection. So thank you for listening, and does anyone have any questions?
Thank you for the talk. Are there any questions either online or in person? Okay, I'll have a question then. The plots you were showing, what inversion time was that at? I mean, were those simulations assuming some inversion time, or?
Yeah, the inversion time, I believe, was 950.
Okay.
I could actually pull the paper now.
I see. So you're acquiring an inversion time that's close to nulling out CSF.
Yeah. Specifically, it was a TI effective, which would be the TI where you had like a max value.
Mm-hmm.
So yeah.
Great. And do you know like where on which of those excitations are you going through the center of K space? Are you doing centric encoding or linear encoding?
Yeah, so the paper that we got this from was actually focused on optimizing the K space.
Mm-hmm.
So they recommended that we get the 88th pulse, but that they use that to calculate the optimal TI, right? So I guess in this case the optimal would be actually the 0 on the Kth readout, because I did the simulation with the optimal TI, because the TI effective is actually the time until the max readout.
Okay, so you're saying in the paper they recommended taking sort of the middle echo, like the 88th echo would probably be right about in the middle.
Right about in the center. Yeah.
Okay. All right. So linear encoding. Okay.
Yeah.
Any other questions? Okay, great. Good job. And ironically, we have 3 talks on compressed sensing, so we're not being very sparse with compressed sensing, but that's the way it is. Some years there's more or less, and this just happened to be a year with a lot of compressed sensing.
We hope we'll get another perspective.
Sure. Yeah, no, that's fine. It's
hopefully it will be like interesting and people are not going to be bored.
No, I wasn't implying that at all.
Oh no, I didn't mean that.
Okay. Yeah, so we'll be going over the principles of compressed sensing and how we can apply it to MRI. We'll be going over our simulation results. We'll be simulating the reconstruction of understandable case-based data as if it was like the actual MRI sense, taking from case-based and getting our images that way. Is it not working? Okay. So for the outline, we're gonna go over MRI just really quick and talk about the need for speeding it up. Then we're gonna be talking about case-based sampling and having enough data, how much data we need, compressed sensing and a brief intro, reconstruction, undersampling MRI scans, reconstruction results, comparison, and the last one, I'm sorry, robustness and limitations. MRI is a very powerful tool. It has benefits over CT scanning, especially in that we can contrast different types of tissue. The one caveat to this is that it's just an inherently slow process. This brings up some areas of concern, namely people with like physiological impairments, or babies, for example, that can't stay still, and also maybe a business perspective, patient throughput. One solution that got a lot of popularity is the idea of compressive sensing, and the idea behind this hinges off of the fact that natural images are a very small subset in a pixel space. That is to say that they're very compressible. That like a picture of a face is somewhat similar to pictures of other faces, whereas like in the vastness of the pixel space, a random noise is vastly different from these natural images. And this constitutes a convex optimization problem that we can solve so long as three conditions are met. One being the sparsity, the incoherence, and the nonlinearity of the minimization. And so long as those three are met, then we can find such an M vector that would be the minimum solution to this. So to walk through like a classical example with just the Fourier reconstruction, at the bottom right is where we might be accelerating the acquisition of case-based data when we just randomly sample. To do that in our simulations, we show that we take our original image, we take the Fourier transform. The measurement matrix is now our random sampling. We multiply that by FFT, and this is our case-based undersampled. One thing to notice, of course, we saw in previous demonstrations is that the magnitude is much higher in the center. As you can see, it's kind of circular. So there is a need for the incoherence to not say zero out everything in the center. And so if we just do the Fourier transform, the inverse Fourier transform, we get a very like high artifact image that is just not very usable. So in a kind of a just a kind of a dry run through compressed sensing, if F is the matrix that we want that has something that is incoherent to the transform and is nonlinear. The minimization is nonlinear, and so dm and dn, we take the Kronecker product such that we can take the 2D Fourier, or in this case, discrete cosine transform. If we take the Kronecker product of going vertically and horizontally, we can use this as our transform from one space to another. And in doing so, this is some of our work we see. We have a 630 by 630 image, and The issue here is that the Kronecker product is sort of a bottleneck. Doing so creates a very large matrix, and it's very computationally expensive. And as you increase the size, it gets exponentially more time consuming, and so what we see is we had to downsample to 5% and 10%, and following this process, we used half of the case-based data, and the reconstruction shown on the right. They are not great, obviously, because they're very small and pixelated, and this kind of points out the smaller your pixel space, the less compressible your images are. So, but in the early 2000s, what we found is that a lot of optimization algorithms allowed us to perform the same optimization scheme, but on the larger, non-downsized images. So these are still the 630 by 630. So we don't do any downsampling here, but we do compression. And from left to right, you see original. Top left is original. So then next is 60% of the case-based data, 40%, 20%, 10, 5, and 1%. And the reconstructions are shown here. And it's interesting to note that just 1% of the case-based data, the very last image on the bottom right, kind of looks like, maybe you just didn't put your glasses on, but we're using just really, you can hardly see the speckles of the original case-based data. But we could say here possibly that 60 or 40% is still somewhat usable. And that could mean roughly half the time acquiring this case-based data. And just for another reference, this is another picture. The performance, you could say, still is pretty good, 60 to 40%.
Now, in MRI imaging, unlike natural images, data is acquired in actually the frequency space. And then data is taken to the spatial domain. Then a sparsity transform is minimized using sparsity promoting norm. Now, the case-based data has some inherent structure that we know of, and some of my colleagues have talked about that there is a higher density of samples in the middle. So it doesn't make sense to completely just randomly sample, knowing that we have a lot more information in the middle, and that's one of the things that we're gonna uncover. The norm is then minimized with straight based on the acquired data. So what we want to minimize is the L1 norm, which is an approximation, a convex approximation to the L0, which is the ultimate specifying norm in the transform domain. That transformation is either the wavelet or the discrete cosine transform or the total variation. And then we have Y, which is the measurements in case-based in natural images. Usually that like the measurements that we're trying to put the constraint on is in the spatial domain. And then we have the image scan in the spatial domain, and then the underdetermined Fourier transform and sampling. And one of the things that Chris talked about is the Kronecker product and how that is a bottleneck by using the matrices. So to speed up the operation, we transform that into using operators in our iterative reconstruction approach. So this can be then transformed into a minimization problem where that constraint can be transformed for a Lagrangian multiplier. So we're basically minimizing this function, alpha control the contribution of either this or this to the loss function. So which one is more important to you? It's basically a trade-off term. Now the L1, to see why the L1 is specifying transform, we're trying to minimize or match an undetermined equation. You see that usually the minimum with an L1 norm is a sparse solution. That means this is a solution that matches this line. If you're talking about the L2, talking about the circuit, we can see that this solution basically in these components is not sparse. Trivial question, we can handle that in a Q&A. Y straight 9 is the solution in this case. So this is something we can discuss later. Now this is our reconstruction or from the experiment. This is the total T2 volume with 22 scans, 256 by 256. This is the original volume. This is reconstructing using L1 minimization, using the minimization problem I talked about earlier. And in the wavelet domain, this is pseudo inverse L2 inverse Fourier transform. Now, using the iterative process, the whole volume reconstruction took 53 seconds. It's 2.4 seconds per frame. That's the whole thing. So it's quite fast in its reconstruction. Now, we did several set of experiments on different scans and on different noise levels. The first one is using a sampling scheme, using completely random sampling. And this is, we wanted to show how this is not a good approach in sampling the frequency domain, knowing that we have more data in the center. So the first row is the sampling scheme, the second is the inverse, and the third is basically the sparse reconstruction. If we go into densely or high density sampling, so like largely random, you can see that even if we have only 10% of the data, it sells better than 25%. If we have 50% of the data is 25% of the data using that high density in the middle is still much better than, oh, sorry, not much better, is as equivalent as acquiring 75% of the data. Now, when we look at this high density sampling, we're also testing several scans with different complexities to try to see if the complexity of the scan does affect the reconstruction error and quality of the reconstruction. All of the scans are 256 by 256 with a ratio of sampling of 25% of the samples. We see that there is still some variations due to the complexity of the scan, but still the reconstruction quality is not quite significantly different from one scan to the other, judging by the SNR and the mean absolute error. We did also another experiment. We took different noise level to see if the reconstruction, how bad is it going to break after a certain amount of noise. And we see that it doesn't quite break. So the level of noise is quite similar or quite close to the level of noise visually that we get in the original scan. And to be honest, we didn't go any beyond that because I think having more noise than that is the problem is not speeding up the scans probably in the way the scan is being applied anyway. So we did test that on the sagittal also scan just for comparison. And all of these, the noise analysis is done with the high density in the middle. A similar results can be seen here in terms of the SNR and the mean absolute error. The last experiment, we've got a minute at the time, so we're done, almost done, is using the systematic sampling, which is more prone to what an MR actually does. I mean, you do that basically because it's hard to speed up an MRI with like random sampling. It's very hard to implement that. Now, one of the constraints for compressive sensing is that the aliasing or the noise has to be non-coherent. So once you induce coherent noise, then basically you cannot basically use it to an extent to reconstruct. Due to the significant aliasing that we can see. To conclude, in this project, we were able to review the concept behind compressive sensing as it applies to MRI and contrast that to natural images, implement several compressive sensing approaches to MRI in different scan types, different noise levels, and different acquisition conditions. We observed that MRI scans are sparse in the wavelet domain and discrete cosine domain. Hence, these two domains can be used as classifying domain during reconstruction. However, we can also observe, especially if you do a lot of undersampling, that there are a lot of artifacts, and that should be taken carefully when doing any diagnostic for any diagnostic work. These are the references, and these are the list of computational packages that we use for our project. And we'll be more than happy to take questions. Thank you very much.
Great. Any questions either on, oh, please.
This one? Yeah. Okay, so one of the conditions, because compressive sensing, maybe not a lot of people know that compressive sensing, especially if you do it like in the wavelet cosine transform like we did, It's quite similar to denoising, knowing that I have a set or limited set of high coefficients in that element, whether it's the wavelength domain or the discrete cosine. Now, to be able to denoise properly, which is reconstruct the image from undersampled samples, you have to have incoherent noise. Once the noise is coherent with the signal, because when you do the undersampling across a space, every other line, for example, if you're doing that, then you're getting that very signal-based coherent aliasing, which compressive sensing is not designed to overcome. Does that answer your question, or?
All right, any other questions, either online or in person? I had a question. So did you, I wasn't sure. So in your presentation, you'd looked at random sampling. Did you also look at variable density sampling?
Yes. So
okay. Can you show me
all this is with higher density in the middle. So,
okay.
So,
and what was the rate? When you did higher density in the middle, what did that look like? Like what was the sampling pattern?
This is sampling.
Oh, okay. Yeah. So how do I interpret that? That's
okay. So
the bright is that okay? So the density is bigger in the circle.
Yes. So okay. More sampling in the middle.
Got it. Got it.
And then like it dies out. Yeah. So lower sampling on the edges, but higher sampling.
Okay. So it's sort of like a step function.
Yes.
Okay. Did you try like you can slowly varying or?
You can make it. The code we built, tested. Like we did it like by circle. But I would assume, not assume, I should know that if you do the varying sample, it should reduces the Gibbs ripples. So that should provide better reconstruction, but at the same time, the assumption also at the same time would be, it's not very challenging to simulate that decaying function. The only constraint is that we know that it's going to be even harder to do that in an MRI like sampling scheme so you probably can sample really a lot in the middle, sample a lot less outside. You want to see how that's going to look.
Yeah, well, actually, with the variable density spiral, you can sort of, it's actually pretty easy to sort of vary.
Yeah. This part, when we use the band, my assumption is not correct.
Okay, no, very good. So why don't we move on? But thank you again.
Thank you.
Okay, last but not least, we have a talk on pills, which is a type of parallel imaging, and RF Coil Design. So is that in person, online?
We're online.
You guys are online? Okay.
Yes.
Yes, we are online. Is everyone able to hear me?
Yeah, you can go ahead.
All right. So hi everyone, we're group number 7 and we're gonna talk about RF coil design. Particularly, we talk about a method called PILS, partially parallel imaging with localized sensitivities. First, we'll go over a little bit of background. So as we have seen in class, parallel imaging means that we have more than one receiver coils and then each coils can scan the object at the same time to reduce imaging time. And the cons in this is we sometimes have a decrease in SNR depending on the method we use. And some methods, including SMASH, SENSE, or GRAPPA, they all require really accurate knowledge of the sensitivity profile of each component coils at the location of each pixel, which is really difficult to determine. So PILS concept is parallel imaging in localized sensitivity. So PILS is a method that does not require knowledge of the coil sensitivity profiles, and we'll see how. So as we know, the sampling rate has to satisfy the Nyquist limit. And like in figure A right here, your delta KY has to be less than 1 over FOV as we've seen in class. And then in B, you can see that when we undersample, there will be aliasing. So the concept of PILS is that we treat each coil as an analog bandwidth filter, limiting the bandwidth to the YC range, as you can see in figure C right here. And then you can also limit our FOV to YC only.
And then like in D, you can see we have a smaller FOV, means that we have less delta KY to cover, so the imaging time will be faster. Next slide, please. All right. And so here you can see that we have an array of coils, 4 coils, as you can see in figure A, and we'll assume localized sensitivity for each coil, which means that each coil only sees a part of the full image. And each of the coils you can see here has a range, sensitivity range of YC. So we have a limit here, that is YI, the FOV that we want to image, it cannot be less than the range of YC, because if it's less than that, then we break the Nyquist rule and it will result in aliasing. So it means that YC is always less than YI, so that our limit, our acceleration factor is always have to be less than the number of coils we have. Next slide. And here we define the acceleration factor as the speed improvement of the MRI system. We define it by the sampling spacings in case they needed to sample the full HOV over the sampling spacing used in smaller FOV acquisitions. So if you look at this previous image from the previous slide here, our acceleration factor will be delta KY of YI over the delta KY of YC. All right. So for the reconstruction of this method, we first write out the Fourier transform relationship between the signal and the reconstructed image, which is the first equation you see on the screen. And instead of sampling from negative infinity to positive infinity, we limit the integration to only Y0. The Y0 is the center of the coil sensitivity range, as you can see from the figure, to plus and minus YC over 2, which only covers the sensitivity range. So that's the second equation right here. And then we Fourier transform the signal in K-space back to get the image, which is the spin density rho of Y prime right here. And then, as you can see in the Fourier transform function in the third equation, we also have a phi of KY. So the phi of KY is an adjustment phase to recenter the image to the correct position, since we will lose the position as we divide it into 3 little FOVs. And since we assume absolute local sensitivity for each coil, it is really not realistic. And there will be overlapping regions like you see in figure A. So there will be margins of errors, we will produce artifacts, and as you can see later in our presentations.
All right, now we're going to talk about how we use MATLAB to simulate and sort of estimate this PLS method. Next. So first we start with just a basic 200 by 200 pixel modified Sheff-Logan phantom. Then just for this example, we're doing an acceleration of 2, in which case we cut out every other line in KY. So we limit the sampling in K-space. Next. Then to no one's surprise, if you try and reconstruct that image, you get a large amount of aliasing, overlapping of the object with itself. However, as you'll see, and we'll go over how the method, how we perform this method, you can reconstruct the image perfectly, even with that acceleration with PILS. Next. So for our first iteration, we've got a really simple coil sensitivity profile that starts at 0 sensitivity right at its edges, increasing linearly up to 100% sensitivity right in the middle and back down. And on the right shows our coil layout. We have 6 coils, each one covering about 40% of the image in Y. Next. Now for one of the coils, you see how the process works there on the left. First, that object there in the top left is what coil 1 is able to see with its sensitivity. The next image, just to the right of it, is the Fourier transform. So I apologize, it's supposed to be zoomed in, but was not. And then the bottom left, that is the reconstruction using the data that coil was able to obtain. And then using just the general knowledge of where that coil is sensitive to, you can get rid of the aliased portion at the bottom that you know doesn't actually exist. If you repeat that for all 6 of those coils, you can reconstruct that full image with an acceleration of 2, taking half the points in K-space. Next. So we did a little bit of an acceleration analysis on that first iteration. A couple things to point out are looking at about 2.75 acceleration, you begin to see some ring artifacts around the edges, which begins to appear due to a windowing of K-space. One of the limitations of our method is that it slightly weights the lower frequency components over higher ones. So if you have a decimal point acceleration, you get rid of more of the higher frequency lines, which causes a little bit of that ringing. Another more specific to PILS, however, is that aliasing first begins to appear around the 2.6 to 2.7 acceleration if you look at the bounds of where the coil sensitivity regions are. The field of view of each coil is 40% of the original, which theoretically corresponds to a max acceleration of 2.5. The acceleration is slightly higher because the original object doesn't span that whole original field of view, so the theory and simulation here match. Next. For a second iteration, coil sensitivity profile remained the same, but we increased the size of each, or the sensitivity region of each coil, such that there were less coils over that whole image, just 4, each one covering 2/3 of the length. Next. In this case, you see once again what the coil is able to see of the object in the top left, Fourier transform, the reconstruction, and then the trimmed using the knowledge of the coil sensitivity region. And this time, even with that same acceleration, you get quite a bit of aliasing. Now, this is because the field of view of the coils we're using are effectively larger, meaning they require more samples in KY to avoid aliasing. Next. Another acceleration analysis, you see that around 1.6 to 1.7, you start getting those ring artifacts emanating from the bounds of the coil sensitivity regions. Once again, like was explained, this is because of the larger field of view of these fewer coils. Next. All right, and for a third basic iteration, we wanted to do a 2D layout. So we generated a coil sensitivity profile by taking that original linear profile, transposing it and multiplying them together to get it in a 2D format. Then for coil layout, we did 4 coils, 1 in each corner, each covering 67 2/3 of the image in each direction, X and Y. Next. Very same process here. In this case, however, despite the coils taking up 2/3 of the length of the image, just like the last iteration, even with an acceleration of 2, we're able to get no aliasing in the image. This is because we're now cutting out points both in X and Y and covering both of those dimensions with different coils, allowing us to have greater acceleration by removing points in both KX and KY. Next. So, lastly, aliasing in this case, in the 2D case, begins to occur at about 2.25 acceleration. This is because the 2D field of view of each coil is approximately 44% of the full image space, which corresponds to a max acceleration of about 2.27. Next.
Okay, so in part 3, let's take a look at some real world cases. Next, please. So here we have a liver MRI image, and there's a colon adenocarcinoma metastasis in the liver. And we use different acceleration factors to acquire images. So in the image with 2 and 3 acceleration, you can still get a reconstructed image without noticeable aliasing. And in the image with 4 acceleration, aliasing becomes more obvious. And it does really affect the quality of image, but can doctors still make a correct diagnosis from this image, at least for colon adenocarcinoma metastasis? In my opinion, the answer is yes. If there's an emergency or a specific situation that we don't have time to wait for a full-time image and the doctor can make a right diagnosis from the image with aliasing, then this aliasing is acceptable. However, in an image with an 9 acceleration, the aliasing is too severe for the doctor to make a correct diagnosis. So this aliasing is unacceptable. So next. So let's take a look at another example. This MRI image shows the injury to deep deltoid ligament of the ankle. The fiber discontinuity making it a grade 3 tier. Likewise, we can get nice reconstruction image with the image with 2 and 3 acceleration, and the aliasing shows in the reconstruct image with an square acceleration. And also we can get the same conclusion that this image with aliasing won't cause diagnosis issues. However, the image with 9 acceleration, even though there is a severe aliasing in the reconstructed image, but maybe the doctor can still make a right diagnosis because we still can see that discontinuity in the image. But in this case, the quality of this image is pretty bad, so we're better not to use this image. So next, please. So the last example is an extreme case of insertion tendinopathy of the Achilles tendon. And in this case, we also get the same conclusion. So next, please. So in conclusion, when should we use the PI acceleration? First of all, in a situation that we need acceptable image for patients who cannot stay still for a long time. And secondly, in emergency of specific situations, which the patients have to be diagnosed in a short time. So and remember that how fast for imaging and how much aliasing is acceptable depend on whether the doctor can make right diagnosis. So if we can achieve our desired voxel resolution and spatial coverage in a reasonable time frame without using parallel imaging, then we're probably better not to use it.
So in this last part of the section and last part of the whole presentation, we're going to delve deeper in the coil analysis by considering the different parameters that affect it and the advancements in the field. So coil analysis is highly dependent on the application and the MRI target, and thus different types are used for each application. It's also dependent on the strength of the field, the coil radius, and the geometry of the coils, resulting in different optimal coil radii. Geometric and mechanical factors such as the coil losses, the decay area, the spacing, the overlapping of the coils are also important. Thus, the quality factor Q is used to compare the efficiency of the coil loops. The filling factor eta is used to maximize SNR. Choosing a coil that fits closer to the region of interest, you potentially gain better images, and this will be the hypothesis for the last results, for the last experiments done. So in these experiments, we designed 4 different coils that wrap tightly around the target image, with each of them imaging the lighted region. The reconstructed images the reconstructed image of the lower right coil, which has almost half the size of the 2D pill coils, can approximately image 1/3 of the phantom. So the final reconstructed images for the wrapped coil on the right and the comparison of the 2D pills on the left is drawn. The wrapped coils present better reconstruction of the image in terms of both aliasing and noisy and without noise, and better representing of the small features and less artifacts. The disadvantage of the wrapped coils as presented in this experiment are the uneven lightning in some areas, particularly in the middle. You can see basically like a rectangular, but this is a result of the simplicity of the test and the sum of squares for reconstruction method. So about the advancements in the imaging field, a lot of them target the RF coil design. Metamaterial coils tackle high specific absorption rate levels and image inhomogeneities, aiming to achieve optimal and effective material properties such as permeability. Big data and digitization advances aim to develop more effective MR scanner systems by letting more than 128 channels at the time of writing, which is a limitation to the current machines. Optical and wireless technology simplifies coil handling by the operator. Wireless coils advance safety by eliminating balloons, offering convenience and ease of operation. Multi-tuned RF coil systems allow for the examiner to do metabolic investigations without changing the RF coils or replacing the subject of interest. Lastly, one-size-fits-all RF coils are targeted through flexible RF coil design, which we'll look at a bit more. So the flexible adaptable RF coils are sensitive and comfortable. They're adaptable to the patient's anatomy and minimize compromises in sensitivity. They allow for flexion of joints and promise enhanced workflow, while it has some great pediatric applications where conventional rigid arrays are proven sufficient. So these are references. Thank you very much for your time, your attention, and we're open for questions.
Great, thank you. Are there any questions from online or in class? Okay, I had a question. So for the pills, did you guys just do a sum of squares recon of the different coils? How did you
So
different coils? Was it just sum of squares?
yeah, it was really just a really basic, just layer them together, sum of squares. Yeah.
Okay. And then when you did the fractional sampling rate, like 2.7 acceleration, did you oversample and then go to where you could have uniform sampling to get exactly fractional, or did you sort of round off and then just skip, not do exactly fractional sampling?
Good question. We actually went through a few different methods to get that to work. And the one we landed on that was most computationally efficient, let us do it easily, was we would just do a normal Fourier transform. Like, let's take a simple example, 2.5 acceleration, in which case we would take the middle 50% of the region. So if we were just working in KY, it would be that 50% band down the middle. And those would be, we would retain every other line in that middle 50%. And then the outer 25 and 25%, we would retain every third. And this was a close approximation, would result in aliasing at the correct level of acceleration, but also cause a little bit of ringing since we're getting rid of more of the higher frequency components.
I see. So you sort of did a non-uniform subsample.
Yeah.
Great. Okay. Any other questions? Okay, great job, everyone. I think very nice presentations from all the groups. So I think we're really happy to see everyone dive a little deeper into the material. And so this concludes this quarter. So thank you everyone for attending. Obviously, if you have any follow-up questions or anything, just let us know. And we hope to see you in their future class. I think Professor McVeigh is teaching 280B in the spring, right? Yeah. And then 278, as we mentioned before, is being taught next quarter. And so that's MRI and CT lab. So good luck with the rest of your exams and studies, and we'll see you around. Okay.